All right, and again, welcome everyone. Um, Let us go ahead and get started. So, hello, thanks for joining us today. This is a CNS Tutorial SA5 from synapses to behavior using open data, tools, and models from the Allen Institute in Computational Neuroscience. So it's great to have you all here with us today. And I see some new names today. We had another day of tutorials yesterday. So just for the sake of the new people who are joining us, we'll have a bit of an introduction first, and then we'll talk about the details of each of the data sets. So first things first, of course, we all are used to Zoom by now, but just as a reminder, this is a webinar, a Zoom webinar. And so as attendees, you can interact with us and with each other using chat and Q&A. We just ask that you use chat for general conversation. Try not to post questions there. It's much better to post questions in Q&A. Otherwise, there is a chance that questions can be lost. So yeah, and also keep track of who you're sending a message to. It can be all panelists or all panelists and attendees. Depending on that, different groups of people will see your posts in the chat. And then in the Q&A, please post your questions to the panelists, to the people who are presenting. We are trying to leave some time at the end of each presentation to answer questions. And then if there is something really important that needs to be answered right away, the speaker may choose to stop and answer, but otherwise questions will be answered at the end. All right, so I hope this is all clear. Thanks again. And then just a few words about who we are and what we are discussing here today. So we are talking about the data and tools from the Allen Institute, which is a non-profit biomedical research organization that was established in 2003 by Paul G. Allen. So we are in the beautiful South Lake Union neighborhood of Seattle. And the Allen Institute is a big organization. We have over 500 employees now, and it consists of multiple divisions. There are divisions that work in brain science or in neuroscience, as well as divisions that work in cell biology, in immunology, and there is also a funding group called the Paul G. Allen Frontiers Group. So the whole idea for the Institute is that we focus on hard problems, complexity, and foundational questions in biology using these approaches of big, team, and open science. And as a result, we produce comprehensive data sets and tools and knowledge, which we share with the community. And that's really why we're here today, to share what we have with all of you. So a few words about the core principles of team science, big science, and open science. As many of you know, the Allen Institute is somewhat different from the regular academic research lab, specifically for the reasons that we want to pursue this team and big science approaches. It's complementary to the regular research approaches in the university labs, which are usually a specific question or hypothesis driven. We usually pursue questions that are more about obtaining a complete full picture of some kind of facet of the system that we are studying. And so we use the team science approach for that, working in multiple interdisciplinary teams that come together to address the common goal. So we usually try to do something that is very hard to do in a regular university setting, to produce large-scale data sets, pursue large-scale projects with robust, massive data with high-quality controls. And ultimately, this data are made available to the community, where the university type, individual type, lab type research can be done using those data, right? And that brings us to the point of about open science, which is really a foundational principle for us. We share all our resources, the data, the tools, the knowledge freely online at brainmap.org. For software tools, we share them usually on GitHub. And so all of that is made publicly available, typically 1, 2, 3 years before any publication of our own. And so, yeah, this is the big guiding principle and something that we believe is really important for the community. So the Institute was started in 2003. And in the beginning, it focused on gene expression atlases. It sort of became quite well known for example, the Allen mouse brain atlas, which is widely used in the community. But in the last decade, we added to our portfolio a diverse set of systems neuroscience projects. And starting in about 2015, new data sets started being produced and shared with the public. So we had a data set on the Allen cell types database with the properties of cells in the mouse brain and in the human brain as well. Then we had a release of the Allen Brain Observatory, the information, the data from in vivo recordings from the brain, from individual cells using 2-photon calcium imaging. More recently, we had a similar data set with electrophysiological recordings using neuropixels probes. We have models that we are sharing with the community, synaptic physiology using multi-patch characterization of connectivity and synaptic properties. More recently, characterization of connectivity in the brain using electron microscopy. And the latest addition to this family of data sets is the visual behavior data set that you will hear about today with the optical physiology recordings from the brain of awake behaving mice. So these stars, they denote the data sets that we are covering in these 2 days of tutorials. Yesterday, we talked about the 2-photon and neuropixels passive viewing brain observatory data, as well as Luke presented the synaptic physiology data set. And today we'll be talking about the cell types, characterization of cell types, and then about this newest data set, the visual behavior, and we'll wrap up with description of models and modeling tools. So also, just to give you a little bit more information about how we operate, there are actually multiple divisions at the Allen Institute doing neuroscience. One of these divisions is, so the name is relatively well known. It's the Allen Institute for Brain Science. And that division has a lot of projects in its portfolio. Perhaps some of the most visible ones are projects that focus on the cell types, characterization of cell types, and connectivity. So sort of the structure of the brain, what is it made of and how it's connected, right? And so for the cell types, there is the characterization using transcriptomics, morphology of cells, and electrophysiology of cells. And as you will hear today, there is actually a triple modality characterization of individual cells. On the connectivity side, there is a synaptic physiology and electron microscopy ultrastructure and other methods to characterize the connectivity. But also something that I don't even show on this slide, there are things like transgenic mice, which are widely used in the community and also produced by this division. On the other hand, another newer division is called Mindscope. So for example, I'm from Mindscope. And it's another division at the Allen Institute that also deals with neuroscience. And in this division, we focus more on recordings in vivo and on modeling, network modeling. So on the previous slide, one component of characterization of cells and synapses is also modeling of individual cells and synapses. Here, we try to put it together into network models and connect it with these experimental recordings, which is a big part of the portfolio of Mindscope, using electrophysiology or calcium imaging in vivo in awake mice, either passively sensing or behaving. All right, so this was our schedule yesterday. We talked about synaptic physiology and 2-photon and neuropixels recordings in awake mice. And today, after this introduction, Nathan will tell us about PatchSeq, characterization of individual cells, of their morphology, electrophysiology, and transcriptomics. Then we'll have a short break. And after that, Alex will tell us about the visual behavior and 2-photon recordings in these behaving mice. And then we will have the last session about biorealistic modeling and modeling tools with me and Kael. All right, and finally, you can find all the materials to follow along with these tutorials here on this link. Please feel free and follow in real time, or just take a look at those materials and maybe go through them later. They are all there, open for you. So everything is in Jupyter Notebooks, freely available. Okay, so thanks again. We are very happy to have you here, and let us get started. And now Nathan will tell us about the PatchSeq dataset. Thank you.
All right. All right. Yeah, thanks, Anton, for that overview. So my name is Nathan Gouwens. I'm at the Allen Institute for Brain Science, and I'm going to be talking today about our PatchSeq resources, and I will explain what PatchSeq is, what types of data we're collecting, and the ways you can access it, including the newest way, which is a web application. I don't think this is the most recent data set released by the Allen Institute, since most of the data was made public last year, but I do think it is the most recent web application that we have released, which was, I think, just, I guess technically it's 2 months ago now. Yeah, so let's get started. So at the Allen Institute for Brain Science, we are interested in characterizing and classifying neurons into cell types. And so these are some of the ways that historically and even up to now, we try to put cells into different types. And we want to do that because we want to understand the sort of fundamental components that make up neural circuits. And you could, in some sense, every neuron is unique as to itself, but it is a very helpful conceptual abstraction to break things into somewhat more generalized types to figure out how they connect with each other. And so we can all be sort of talking about the same components of these circuits. So, initially, the most common way to classify cells into types was based on how they looked, on their morphology. Their morphology, where they're putting their processes, is often a proxy for connectivity. So connectivity has also certainly been used and we can now often measure that directly. Another common way that starts to relate to what a neuron might be doing in a circuit is its physiological properties. And so that can be intrinsic physiological properties, which is what we'll be talking about today, but also, responses to sensory stimuli and during behavior and things like that. And then another very common set of tools for identifying cell types are molecular markers. So staining for something like NDNF or VIP identifies sort of subsets of cells that often are mutually exclusive with each other. And so those are often quite useful methods for breaking cells apart into different categories. Now, cell typing has really, over the past few years, been sort of amped up by the widespread adoption of single cell transcriptomics. And so with single cell transcriptomics, you take a piece of tissue, you dissociate it into individual cells, and then you can reverse transcribe and amplify the genes currently being expressed by individual cells at a time. And this gives you a very high dimensional readout of all the genes that cell is currently expressing, and also has been really amped up in terms of the throughput. So you can do many, many cells. And so you can get a lot of data, both in terms of number of cells and in terms of dimensions you're measuring, which have been proven quite useful in terms of defining cell types. So you can take that high dimensional data and collapse it using some kind of two-dimensional embedding for visualization, like we're showing here. This is a TSNE. And you can see there's a lot of structure to this data. So different major cell classes that we are very familiar with: inhibitory GABAergic neurons, glutamatergic, excitatory neurons fall onto different islands or continents within this two-dimensional projection. Non-neuronals are on their own, non-neuronal cells are on their own. And then there's finer structure as well. So if you use these to try to determine clusters in an unsupervised way, you find that those also, go together, and some of them are closer to others. And this tends to follow relationships that we already understand. So another way of visualizing this data is with a hierarchical tree. And as I just said, this kind of reflects the sort of previous understanding we had of these types in terms of, again, like one of the major splits you see with the neurons is between GABAergic neurons and glutamatergic neurons. And then within the GABAergic neurons, there are classes defined by major molecular markers such as parvalbumin, somatostatin, VIP, LAMP5. And among the glutamatergic neurons, they often fall into different classes defined by their projection targets. So telencephalic, corticothalamic, and also the cortical layer within they reside. Sorry, I forgot to say that this particular data set is focused on the cortex of the mouse. So so it kind of recapitulates these major divisions that we've already observed with other experimental modalities, but we also find that there is a sort of additional structure at a finer level. And so we want to understand how meaningful is that sort of more granular subdivision. And so another way of kind of asking that question is, so we've got all these transcriptomic types defined from just purely this transcriptomic data set. How well do these types correspond to other phenotypes that we know about and care about? So for example, in some cases, it's actually quite clear. So you sometimes have a particular type like this parvalbumin Viper2 type, which expresses a fairly unusual combination of markers, genetic markers, and also corresponds to quite a specific cell type. In this case, it's the chandelier cells, which are fast spiking, have a quite unusual and particular morphology and particular connectivity. And so that's kind of an easy connection between the transcriptomic type and another well-established type. But in other cases, it's less clear. For example, among these SST neurons, there's a variety of Martinati cells, maybe some or all of them, well, probably not all of them, but many of them might correspond with what has been called the Martinati cell type. Or in other cases, you have something that's over here in the LAMP5 subclass, but it expresses things like the chandelier cells like NKX 2.1 or LHX6, which is often more expressed by parvalbumin and somatostatin neurons. Is that a neurogliaform like the other LAMP5 cells, or is that some kind of chandelier cell because it also expresses NKX 2.1? So we want to get a better sense of characterizing these transcriptomically defined types, and then also refining this taxonomy based on those other modalities that we can measure from these cells. So that's why at the Allen Institute, we have tried to establish a kind of large-scale pipeline to generate what we call this PatchSeq data. So PatchSeq was developed by a couple different labs, and the idea is to collect single-cell transcriptomic data along with other properties that we can measure using slice whole cell electrophysiology. So it starts out where you are targeting a particular cell in a brain slice. So the data set we've collected has focused on inhibitory neurons, or that I'll be talking about today at least, has been focused on inhibitory neurons in mouse visual cortex. And we make use for that of transgenic lines that can label particular populations of cells so that we can target our electrodes at those particular cells and make sure that we collect sort of a comprehensive data set from these cells. But this technique can be used for unlabeled cells as well. In fact, we do that. We use human neurosurgical tissue, slice that and record from it in the same way as we do in the mouse. There, obviously, we don't have genetic labels, but we can still collect these types of data from human tissue as well, which is obviously very useful. Okay, so you have a particular cell you're interested in targeting, and you have your recording pipette with an electrode inside, and it's filled with a particular with a dye so that you can fill the cell and visualize it. So you approach the cell, you establish a high resistance seal on the cell membrane, and then you rupture the seal beneath the tip of the pipette to go whole cell. That causes the cell to be filled with this dye. And also allows electrical access to the interior of the cell so that you can do things like run electrophysiological protocols and measure things like the spiking characteristics and intrinsic electrophysiological properties of these cells. After the recording, you apply suction and take up some of the contents of the cell, including the nucleus if you can get it, and then pull off a nucleated patch. And so then your electrode has this has the mRNA inside that was expressed by the cell, as well as the nucleus, which also has a bunch of mRNA. And you can take that and do the same kind of transcriptomic processing and analysis that we do with the dissociated cells. And then the cell is left in the tissue filled with dye, which we can then visualize and then reconstruct its three-dimensional morphology. So you end up with three data modalities from this experiment. So you have your electrophysiology that you collected first. You have your transcriptomic information that you've collected by pulling out the cell contents. And then you have your morphological information from visualizing and reconstructing the cell. Okay, so first we're going to talk about the gene expression data that you get. And so, this is not as high throughput of a technique as kind of traditional single cell transcriptomics, where you just take a piece of tissue and dissociate it and then do all those cells. This requires the labor of, patching individual cells and then extracting its contents. So we've used the larger data set as a benchmark for typing the cells that we've collected with PatchSeq. And so what this is showing is the expression of different marker genes, comparing the dissociated cells, which is within each column on the left, and then the PatchSeq cells, which is within each column on the right. And we've used that data to classify our PatchSeq cells into these different transcriptomic types, which are the different columns. And you can see kind of just overall, I won't get into detail here, that in many cases, the PatchSeq cells are expressing a very similar pattern of markers as the reference dissociated cell data set. So, like for this particular type, it's expressing PLCH2, it's not expressing LSP1, and so on. So there are a couple, oh, sorry, Luke, is there a question? Yep.
Yeah. How do you know that the mRNA sample is representative of all mRNA within the cell?
Yeah, you don't, and it's probably not. We've actually done some analysis. So like with the dissociated cells, you can dissociate entire cells. You can also just dissociate the nucleus, where you miss out on some of the cytoplasmic RNA. And if you look at basically like genes that we kind of know are in the cytoplasm, you see that you get the most of those genes when you're doing just the dissociated whole cells. You get the least of those when you do the dissociated nuclei, and the PatchSeq is actually kind of in the middle. So we're pulling out this nucleated patch. We end up with, kind of maybe enriching for more of the nuclear, associated or close to the nucleus type of genes. But you do get a lot of genes. You definitely miss out on some of the genes, and we think you tend to miss out on probably the more cytoplasmically distributed genes. But there's enough data basically that you can still pretty reliably make a cell type call. But in most of the genes, you do see, expression across the data set. So, okay, so yeah, so we're able to put our PatchSeq cells into transcriptomic types. But if we were just interested in transcriptomic information, we would just do the dissociative technique. So obviously the thing we care about is looking at their other data modalities as well. So here is just showing some example some example electrophysiological responses from different transcriptomic types. The different transcriptomic types are in these different colors. You can see that since we're looking at inhibitory interneurons in the cortex, there's a variety of firing patterns that you can see, from this sort of late spiking firing to, accommodating or adapting firing to fast spiking and, irregular firing. And something you can just see from these handful of examples, but certainly holds up when you look at the larger data set, is that there's quite a bit of diversity across transcriptomic types, but there is more homogeneity within a particular transcriptomic type. So, most of the cells from, the parvalbumin TPPG type look very similar to each other, as do the ones from the SST HPSC CBLN4 type. That doesn't mean that there are not different transcriptomic types that look like each other as well. Many of the parvalbumin types have very similar fast spiking behavior, but we do see this consistency within a transcriptomic type that sort of, in some senses, helps us validate that that's a real thing. Okay, so then you can also look at their morphologies, which you see kind of a similar story. So there's a variety of individual cell morphologies. Most, with these interneurons, it's most clearly seen in their axonal arborization, which is what's shown in the lighter colors in these plots. But again, you tend to see a more consistent set of morphologies within a transcriptomic type than you do across the entire data set. So, these SST CALB2 PD-LIM5 cells have a strong innervation of layer 1. You see, these bipolar morphologies among different VIP types, although there are definitely some differences, again, like with innervation of layer 1, and then a variety of neurogliforms, that sort of thing. And then the chandelier cells over here, like I mentioned before. Okay, so we have these 3 data modalities. We can look at it just from a transcriptomic type perspective, but we also wanted to use these 3 modalities together to define types maybe more where at least all 3 of those modalities are coherent and consistent with each other, because we feel like a cell type is most useful when it's sort of aligned in multiple dimensions, where it is similar and different, like the type system is similar and different from each other in multiple modalities rather than just a single one. So we took our MET data, and I don't have time really to get into the details of this technique, but you can certainly look at our paper published last year for that information. But basically, we'll look to see how much the different MET, so morphology, electrophysiology, transcriptomic combinations overlapped with each other. We displayed that kind of cross-mapping probabilities as a graph, and then used a community detection algorithm to identify the clumps of closely related MET combinations within that graph. And those are what we called the MET types. And so this is a correspondence between the original T types and the MET types. And we find that in many cases, what we see is sort of a consolidation. So you have several related T types that go into a single MET type, which has consistent electrophysiological and morphological properties. So in this case, it's kind of consolidating several LAMP5 types. Down here, you can see other consolidation with different parvalbumin types. So that's kind of a common motif when comparing the T types to the MET types. There are certainly cases where you have a kind of a one-to-one correspondence. So these SST Chodl cells, which are SST expressing but long range projecting inhibitory cells in the cortex, those correspond just to a single type, which doesn't have cells from any other transcriptomic type. Similarly with the chandelier cells down here, and with a couple other ones, like there's this more unusual deep parvalbumin basket cell that falls into its own type. And you can see a couple others. What we see less often is sort of an individual transcriptomic type being split up kind of consistently into 2 cleaner types, although you do occasionally see that. So for example, the SST CALB2 PD-LIM5 type actually does split pretty well into this SST MET3 and 4, which have relatively little combination from other T types. And that's because this T type is the one that I mentioned that has a strong layer 1 innervation, but it actually is found both in layer 2, 3, as well as in layer 5. And so what the MET type definition does is actually split those 2 into different populations. So you have the CALB2 PD-LIM5s in layer 2, 3, and the ones in layer 5 as 2 different MET types. So this is just kind of an overview of those MET types. So we've defined 28 of them in all, more of them among SST, fewer of them in these other major inhibitory subclasses. But this is just kind of an overview of their morphologies, their electrophysiology, their action potential shape, their laminar distributions, that kind of thing. So just kind of zooming in on the LAMP5 MET types, you can see that we've defined 2 there. There's this kind of one that consolidated several of the types, and then this more unusual one, which is that LHX6 expressing type that I mentioned toward the beginning of the talk. We do find that it is also a neurogliform cell. It has this late spiking phenotype. It's not fast spiking. It has that neurogliform type morphology, but it is found deep in layer 5 and layer 6. And you can see that where it falls, where these cells fall on the overall transcriptomic landscape, which is being shown here with a UMAP projection, as well as where it falls in the electrophysiology landscape, which is another UMAP projection just based on electrophysiological features. But it doesn't, just because they fall into a single type doesn't mean they're completely homogeneous. We actually think that this LAMP met type 1 is more of a continuously varying population. And it's interesting that that continuous variation seems to be aligned with these different modalities. So what I'm showing is cells from that particular type. The top plot is showing their level of NPY expression. And you can see that the cells on the left side of this plot have high NPY expression, and cells on the right have AY expression. And we've did this dimension by using principal component analysis on the LAMP5 class and looking at the different dimensions. And this is the one that actually corresponds to this difference. So maybe it's not surprising that NPY expression varies with a transcriptomic dimension, but we also see that the width to height ratio of these cells varies along this dimension as well. So the ones with a lower width to height ratio, so they look more ball-like, like this one, tend to have high NPY expression, and the ones on the right, which are wider than they are tall, tend to have low NPY expression. And that also varies with an electrophysiological property, where the ones that are more ball-like and have a high NPY expression also have a longer delay to their first spike than the ones on the other end of this continuum. So this is an example of the kinds of things you can try to glean from this triple modality data set, since we have all three of these types of data from the same cells. Okay, so that's where I'm going to stop with the slides, and now I'm going to switch over to talking about how you can access these data and use some of the tools that we've developed at the Allen Institute to analyze them on your own. So I'm going to switch over to my browser. Okay, so this is the main Allen Brain Map, portal.brain-map.org site. It's exciting to talk about this new web application that we have for browsing the mouse PatchSeq data. And so you can find that right here under new data and tools, explore the mouse PatchSeq data. It's also linked from the GitHub site for this tutorial. So if you click that, you are launched into this web application where you can start to browse through the different PatchSeq cells that we have publicly released as part of this inhibitory mouse data set. You can see that each row is a different PatchSeq cell. We have like the first piece of information is the specimen ID, which is the particular identifier for that cell, which you can use to collect data from that's located in different places, which I'll talk about. You have some kind of metadata about the cell, such as the brain region it's from, its different type assignments, if it has them, the layer that it was recorded from, and the genotype of the mouse it was collected from. We also show the different T-types or MET types that particular cell has been assigned to. Not all the cells have morphologies. I should have mentioned that earlier. So there's about 4,000 or so cells in this data set, and between 400 and 500 of them actually have morphologies. So those are the ones that actually have an MET type assignment. There are so the way we visualize those T-type assignments is with this kind of wheel where it shows basically the structure of this kind of simplified tree. So basically, is it an MGE neuron or a CGE neuron or an MGE neuron? Is it VIP or LAMP5 or SNCG or PV VIP? And then which is the particular type it's assigned to? I'll go through that in more detail when I talk about how you can filter the data, but that's the type assignment. And then the size of that circle represents our confidence in that assignment. So this one is quite confident that it's being assigned to this particular type with an 85% probability, but there is some probability that it's being assigned to its neighboring type. And so that's something important to keep in mind when analyzing the data. So we also are showing example electrophysiological traces, and you can see how the cell responds to different step current injections, as well as a blow up of its initial action potential shape. And then here we're showing the morphological reconstructions of the dendrite in the darker blue and the axon in the lighter blue. You can see it like as its actual, full reconstruction, as well as the depth profile of the dendrite and axon as kind of a more simplified view, and then where its soma is with the red dot. And then again, you can see the MET assignment. This is not a probabilistic assignment, so it's really just showing the dot of where that falls. Okay, you can also see individual properties for these cells that we've put into this web application. So here we're just showing basically the properties that were already being visualized, but you can click this button to change which properties you want to see in the table. So for example, with electrophysiology, maybe we want to see what its upstroke and downstroke ratio are, or maybe for morphology, we want to look at the total length of the axon. And so you can click those. You can see that the table updates behind, and then you have the actual values of those properties shown in the table. And you can sort by those. You can, I'm not going to do that right now, but you can change you can sort by multiple factors at a time to find, maybe you want to really find the ones with the narrowest action potential or something like that, or the longest axons. Okay, I'm going to switch back to the thumbnail view. Now, this is showing basically all the cells. I mean, it's showing only 50 at a time, but we have access. If we want to page through, we can see all of the different cells. But we also have this filtering system so that you can zoom in on just the cells that you're interested in. And so this transcriptomic tree, again, is represented in the circular wheel, and you can use that to filter down the specimen. So if we want just the MGE neurons, we can click that section of this circle, and that will show us now we have all these SST neurons initially, and then eventually it'll go to the PV neurons filter down, and you can see that that's reduced the number of cells that we have selected. We can then maybe just focus on the SST neurons, and then even pick a particular type. Okay, and then I'm going to put that out of the way so you can see. And so now if we scroll through these neurons, you can see that these are cells that have kind of a more, unusual, or not more unusual, but a more particular morphology. They're often found in like upper layer 5 or layer 4. They have a relatively dense innervation around layer 5 or layer 4. You could even go and look at a, filter. You can look at a related type and see how it actually has a somewhat different morphology, where this now doesn't have really that kind of tuft in layer 1 as much. It has this, but it does have this dense layer 4 innervation in many cases. Okay, well, and like I said before, this actually shows everything that has some assignment to this particular type, but maybe we want to just look at the ones that we're most confident in. So you can use these filters just to pick ones that are between 90% and roughly 90% and 100% confident in that particular type. So that narrows us down to 105 cells. And then maybe we just want to look at the ones in layer 4. So you can click on that, and now we're down to just those 49 cells. And you can see that the morphologies and things become even more consistent when you are even more specific in your filters. Okay, so let's say you've gone through and identified things that a particular set of cells that you care about. If we want to expand the capabilities of this Web Explorer, but for now, what you would do is you'd click this download button to get a list of specimen IDs for these cells, which then you could use for additional analysis of the kind I'm about to show you. Okay, so this is a new application. We definitely want to expand its capabilities, but it is primarily at this point for browsing the data set and filtering to particular subsets of cells based on their properties and their metadata. If you want to actually work with the data yourself, you would go to a different page where we link out to the different archives where these data are actually stored. So if you want to do that, you would go to the, again, the main brain map page, and you could go to Explore Cell Taxonomies, and then you scroll down to this multimodal characterization of mouse and human neurons and click Learn More and Download Data. So this is an overview of the different multimodal PatchSeq data sets that we've collected. So again, there's the neurons in the mouse primary visual cortex that we've just been talking about, and we'll get into that more in a second. But I wanted to mention there's also data from glutamatergic neurons in layer 2 and 3 of the human cortex, which you can also download and analyze. And then we've also tried to make information about this PatchSeq protocol itself, at least the way that we've implemented it at the Institute, available. So we have a bioRxiv preprint about that, as well as a PatchSeq tools GitHub page that you might want to investigate if you're interested in doing this kind of thing yourself. Okay, so if you want to analyze these mouse neurons that we've just been talking about, you can click Learn More. There's a little bit more information about those things, as well as links to the publications and the bioRxiv preprint. And then the main thing, if you want to actually access the data, is this downloads table, where you have links to different files or sets of files with those different raw data, basically. So there's the first thing is this table of metadata, which has information about the cell and its type assignments and things like that. There's also just a download instructions in case you know, as a reference for actually getting these things. There's a file manifest, which has the URLs for the data files for each individual cell across the different archives. And then there's direct links to those archives here for the gene expression data, the electrophysiology data, and the morphological reconstructions. Okay, so now I'm going to switch over to my Jupyter notebook to talk about using some of the software packages to analyze those data. So I've got this Jupyter notebook going. I've already imported my kind of basic NumPy and Pandas libraries for working with the data. So the first thing I'm going to do is, so I've downloaded that metadata file that I mentioned already, and I'm just going to load it and look at it. You can see that it's this has things like the cell specimen ID, which we talked about. That's that particular cell identifier. It has a longer name for the cell, as well as which brain hemisphere, structure, things like that. And you can also see at the end, it has these T-type assignments and MET-type assignments. I'm also going to load up the file manifest. So again, that's just a CSV that I've downloaded from that page. And here we have different file IDs and file names, but these are also findable by the cell specimen ID. And then here are the URLs to actually access those particular data files. All right, so, so the transcriptomic data is actually quite large. So I've already downloaded that and even read it into this notebook. So I'm not going to do that right now, but this is basically, if you download that file, this is how you would read it in. It's basically expression of individual genes as counts per million. So it's somewhat normalized to the total number of reads from that cell. And in this file, each row is a gene and each column is a cell. So let's just look at what that looks like. So again, this is these are the different genes as the rows. These are the different cells with a different transcriptomic identifier. And then these are those CPM values. So you can see that some of these cells are expressing this particular gene and others are not. And there's about 46,000 genes and about 4,400 cells in this data set. Okay, so one thing we can do with this whole genomic data set is to create that two-dimensional embedding like the ones I was showing in the slides. And so we'll do that by transforming the data and then using the UMAP algorithm to create that two-dimensional embedding for just a set of differentially expressed genes. And so those are genes that I've already identified. Actually used a different program or a different package called Scratch HiCat to do that. But it's about 4,000 differentially expressed marker genes for this data set. So again, I'm not going to actually calculate that right now. I've already pre-calculated that, but this is how you do it. So you'd read in your markers and you would, from the gene data, you would just take those genes and then do this UMAP embedding to get your coordinates. I do need to import my plotting libraries, Matplotlib and Seaborn, and then I'm going to just plot those embedding coordinates on a scatter plot. Okay, and so you can see this is what you get. Since I've looked at this before, I know that these different islands correspond to the major inhibitory interneuron subclasses. So SST are here, parvalbumin are down here, LAMP5 are over here. So remember, I downloaded those specimen IDs from the web application from a particular SST transcriptomic type. And so I can load in those specimen IDs and plot them here. So here's what I'm going to do. So I'm going to load in my specimen IDs file. I am going to use the metadata file to get their transcriptomic identifiers. And then I'm going to just plot those particular cells in a different color on my plot with the background of everything being plotted. Okay, so that's what you can see here. So all the cells are in blue, and then just the cells that I downloaded from the web application, those 49 cells, are shown in orange. And you can see that almost all of them are in this relatively specific location on the plot, which corresponds to that particular transcriptomic type. We can also just use, if we don't want to, go to the web application, we can certainly use the metadata file itself just to identify particular cells. So maybe I'm interested in those LAMP5 cells, like I talked about during the slides. So in that case, I can just pull out the ones for, let's say I'm interested in this particular LAMP5 PLCH2-doc5 type. I can get just that metadata, which you can see here. If I scroll over, you can see all of these have that particular transcriptomic type. And then I'm going to do the same kind of plotting, basically pulling out just the cells that are in my restricted metadata set and plot those in orange on the same kind of plot. And now you can see that those are found in a different location over on this LAMP5 island. And in fact, they're even, if you zoomed in, you could see that they're occupying just a particular set or a particular sub-location on that island. You can also just look to see how individual genes look in this embedding. So, MPY is one that we looked at before. So we can look at its log, +1 transformed version of that MPY expression and see how that varies across things. So you can see that's what it looks like here. You can see on the LAMP5 island, there's one end that has high expression, the other end has low MPY expression, but there's certainly plenty of MPY expression in other cells as well. So there's MPY expression among different SST cells, the Chottle cells, and the in some VIP cells, and even a little bit in some parvalbumin cells. Okay, so we've just been talking about transcriptomic data again, but we're interested in cells with all three modalities. So all the cells in the data set do have both transcriptomic and electrophysiological information, but only some of them have morphology. So that's what we're going to do here. We're going to filter for the cells with that particular T-type and with that that have a full neuron reconstruction in the data set. And so that will pull out a subset of cells. And if you look at the metadata for an individual cell, because we're going to start zooming in on just a particular cell, you can see, again, its metadata information like its specimen ID, which we'll need, and where it was recorded from, what kind of mouse it was recorded from, and other properties. Okay, so now we're going to turn and look at electrophysiology. So we're going to first need to get the NWB file that has so NWB, stands for Neurodata Without Borders. It's a standardized file format for electrophysiology, as well as other types of data like optical physiology data. And then we're going to use the Allen Institute's IPFX library to process it. So in normal circumstances, you would use, you could grab the file URL from the manifest and use so these cells are actually located at the Dandy archive. And you could use that URL to download it. I just discovered when I was making this tutorial that there've been some changes at the Dandy archive, so not all of that is working right now, but you can still access those NWB files via the web, but we will resolve it and you'll be able to use those file URLs again in the near future. So right now I'm just going to show you kind of how you would do that. So this is how you would get, we want to pick out just the files that are associated with our particular cell that we're interested in. So we're getting this specimen ID from the metadata, from the cell specimen ID, and then we are using the file manifest to find all the cells that are associated with that cell, and then also the cells that are of file type NWB. And so then if we want to look at the actual file name for that cell, you can see it has this particular name, and you can use this information to find that particular file on the web via Dandy. And you would use another field in here, the URL, to actually get the path to download it. But I've already downloaded it, so we'll just move on and use that downloaded file here. Okay, so I'm importing a couple different things from the IPFX library. So I'm going to create an ephys dataset by loading that NWB file. And then I'm going to basically extract kind of the standard set of electrophysiological features that are assuming that the particular protocol, the standardized protocol that we run in our pipeline has been performed on this set. And I'm also going to make sure I'm not analyzing failed sweeps from that data set. So that's all I'm doing here. So that will load that file and run. And you can see it's analyzing the different types of electrophysiological protocols that have been run on it. The long squares, which are like 1 second long current steps, short squares, which are 3 millisecond long current step, and then ramps, which are slowly increasing current injection. Okay, and so we've got those features then in the outputs of that function. And the main thing we're going to look at initially is these cell features. You can see that this is just a dictionary where we've put the different features that have been calculated from the different protocols under these different keys. We can look just at the long squares. And you can see a variety of information about the cell, such as what its Rio base current was and which sweep was that Rio base on, which of the sweeps actually fired spikes, what's the slope of that FI curve, what's the hyperpolarization induced sag, what's the input resistance, the membrane time constant, things like that. So in this case, I'm going to look at the features of that Rio base sweep, where I can see, for example, it fired 5 spikes per second. I can see what the resting voltage was, which is close to minus 70 millivolts, and more information even about the individual spikes. Maybe I'm interested in the latency because we plotted that earlier in the slides, and I can see that there was about a ½ second latency. So this spike fired about halfway through that 1 second long current pulse. And then I can use this information to actually get that raw electrophysiology data from my NWB file. I'm just using its sweep number, and then I'm just going to plot the time versus voltage from that particular sweep. And you can see this is the response from the cell. The current step turned on around 1 second, and the cells fired around 1.5 seconds. And obviously, you can do a lot more with this sweep than just plot it, but that's what we're going to do for now. Okay, so now this cell also has a morphological reconstruction, which is stored at the brain image library. And so, oops, sorry. And so we will grab that file again from the manifest and download it. So we're going to now get the cells of file type transformed SWC. The transform just means it's been uprighted so that the PIA is on the top and the white matter is on the bottom. So it's not just kind of at a random angle. You can see what that that URL is, and then we're just going to download it. You can see we can do that pretty quickly. And that's the name of the file. I've actually downloaded it twice now. That's why that .1 is there. And then we're going to use a different software package from the Allen Institute called Neuron Morphology, very creative, to load that SWC file. SWC is a file format for expressing kind of where the different points of a 3-dimensional morphological reconstruction actually are in the tree structure of that cell. So we're going to import our thing that loads a morphology from the SWC, and we're also loading our constants that tell us which different compartment types are within that morphology, so we can color them differently. So I'm going to load that from the SWC file, and then just make a relatively quick plot of that reconstruction. So I'm pulling out the different nodes from this morphology for the axon, the dendrite, and the soma so that I can color them differently. I'm getting their X and Y coordinates just so I can make a scatter plot. And then I'm going to have those appear in different colors and use equal aspect ratio so things don't look all squished or squashed. So this is what that neurogliaform cell, this LAMP5 cell, looks like. Again, its soma is this black dot here, the orange is its dendrites, and then the blue is its axonal field. And we can calculate morphological features from that. So, again, in the slides, we were interested in looking at the width versus the height ratio for that particular, for these types of cells. So we can use neuron morphology to calculate different features. So I'll walk through how we're going to kind of do that here. So, neuromorphology is actually meant to be quite flexible in terms of the feature calculation. So there's a little bit of kind of arcaneness, although certainly you can look at the documentation for this package in terms of how we're setting it up. But basically, we want to turn our morphology into like a data structure that can be analyzed. We want to import this general feature extractor object. And then we want to tell it which individual features we want to run. So there is actually a default features that you could import and run. That takes a while, so I'm not going to do that here because we'd just be sitting around waiting. What I really care about is the width versus the height. So what I'm importing is the particular feature called dimension. And so dimension does those different things. I'm also wanting to do it for not only the and not just for like all the different components in the things, but I also want to do it for each individual neuronal compartment. So just the axon, just the dendrite, that sort of thing. So that's what this neurite specializations import is all about. And then I also want to do it in all different dimensions. I want to do it in the X dimension, the Y dimension, which is from pia to white matter, and even in the Z dimension, which is into the thickness of the slice. So what it allows you to do, rather than having different functions to define each one of those things, what this is doing is allowing you to nest those things so that all different combinations of those neurite compartments and coordinate dimensions will be done at the same time. So that's all we're doing here. And so then we create a feature extractor object, we pass it, we register the features we actually want to calculate, and then we actually run that extraction. And so we get that information, which we can then visualize. So after we've done that run, we can look at the results. This is just a dictionary again, and you can see that it's kind of this, hierarchical structured thing. So not only are we calculating the dimensions of all the individual nodes, which is the thing that we care about for this particular application, but it also will calculate that for every point where there's a bifurcation, where the tree actually branches into two daughter branches. Or it will do it just for the like the tips of the tree. So only the endpoints of that tree, because these can vary in interesting ways across different cell types. And it's doing that again for the dendrites, for the axons, and for everything together. So for this width height calculation, we really just are trying to pull out the axons node width and the axons node height. And then we're just going to calculate that ratio and print it here. So you can see that this neuron was about 750 microns wide. It was about 270 microns tall. And so therefore it was close to 3 times wider than it was tall. And so again, if you're interested in the kind of analysis that I was showing before, which I can kind of switch back to. Okay, this is strange. But yeah, so basically what I was talking about before is we'd want to look at things like the MPY expression, that width to height ratio, and the latency to the first spike altogether. So the workflow would be identify your cells of interest, for example, maybe using the web application, maybe using the metadata file, download the NWB files and use the procedure that we went through to calculate the latency to the first action potential for all those cells, download the SWC files and go through the procedure that we just did to calculate the width versus height ratio, and then use that CPM gene expression matrix to calculate the expression of MPY, and then see how all those correspond with each other. Okay, so this, sorry, this isn't quite showing up right, but this data set is the work of a huge number of people. I mean, it's like a whole team of RAs who collect this PatchSeq data. They've gotten extremely good at it and can collect a lot of data in a very short period of time. It's a whole lot of people who are responsible for processing all of that, especially since there's so many different modalities, analyzing it, everything together. And so we're extremely grateful to everyone at the Institute's effort for putting this data set together. Another whole team is responsible for creating the web application and making the data accessible online, as well as developing the software tools that allow other people to make as good use of the data as possible. And then also want to thank the vision and support of our founder, Paul Allen, for making all of this possible. So now I'm happy to take additional questions about the data sets, about using the tools, anything like that. So thank you. All right, I see that there is one question from Yuan Li. Can I hear from you? What is the Institute's plan on into proportions of connections between ME types of neurons, if there's any? It's a good question. I think there's a couple different ways that that could be addressed. So one thing is, like Luke talked about yesterday, there are, there is the direct measurements of synaptic connectivity. I believe that there is work to integrate the PatchSeq method with those recordings as well. So that would be one avenue if that works kind of at scale to do that. Luke, you feel free to talk about that too if you want to. Another way to try to do it is to actually kind of link across data sets. So another data set that we're working on is a large volume electron microscopy data set that's just morphological information, but it's possible that we could, make strong connections between morphological features and that allow us to find cells that we're pretty confident belong to a particular MET type within that electrophysiology data, or sorry, electron microscopy data set, and then look at the connectivity between cells, at least anatomical connectivity between cells that way.
I'll just add a little bit to that. We are doing simultaneous PatchSeq recordings right now. That's a data set that will hopefully come out within the next year. We're still doing a lot of work on that. But as Nathan mentioned, doing those experiments at scale is really difficult. So the project there is going to be focused on a relatively small set of cell types and regions in the cortex.
Yeah, I think we're also interested in methods for, like, with the electron microscopy of, possibly incorporating some kind of identifier of cell type. Hopefully that links to MET type, so that particular processes can be labeled or something like that in the volume, and then you can, make those connections maybe even more directly. But that's definitely still in development.
Hey, Nathan?
Yeah.
So maybe I can also ask a question. Can you tell people a little bit more about the other related data sets that we released previously without the transcriptomics, but just electrophysiology and morphologies? Yeah, and also maybe say a few words about the single cell models that we have there.
Yeah, sure. Yeah, so the kind of earlier iteration of this pipeline did not use the PatchSeq technique, so we didn't extract the transcriptomic contents of the cell, but we did the same thing in terms of the electrophysiologic protocol, the morphological reconstruction, and we were also still targeting cells usually using transcriptomic lines for at least the mouse, of course, just the mouse. So those are available in a different web application called the Cell Types Database. Those are also available. The data are available all publicly, also in SWC format, NWB format. So you can look at those cells as well. There's a lot of cells there too, and both inhibitory and excitatory cells actually there, although we are planning to release excitatory cells for PatchSeq in the near future, we hope. Yeah, and then the other thing we did with those original set of cells is build single cell computational models based on that data. So, and actually a different variety of models. So there were more abstract point neuron models that were built for a large set of the data. And then for cells where we also had a morphology, we built and had enough data. We built biophysically more realistic models, that could be loaded into something like Neuron and used for large scale, biophysically realistic simulations, like ones you'll hear about later today. There's actually different flavors of those even. So the more of them have just the active conductances combined to the soma, but a subset of them also have active conductances in the dendrites as well. And so all of those are available for download from the cell types database.
All right, thank you. Yeah, thank you very much, Nathan. That was great. Well, so I think then we are done for now. If there are no other questions, we probably can break and come back at the end of the hour. So 10 a.m. Pacific time, 1 p.m. Eastern time, 21 minutes from now. So yeah, we'll be back there, and we'll continue with the visual behavior project and then with modeling. All right, should we go ahead and get started?
Yeah, I think so.
Yeah, that's good.
All right. Well, hello, everyone. For those just coming back from the break, we're going to get started on our next tutorial here. My name is Alex Piet. I'm a scientist at the Institute, and I'm going to tell you about our visual behavior project and the data set we just released a few months ago. So I'm going to start off with a few slides, giving an overview about what this project is about and the data set, a couple of our key findings, and then I'm going to go through an interactive notebook that you can find on the GitHub page for this tutorial and show you how to access the data. If you have any questions, please put them in the Q&A and I'll answer them as they come up. So the Visual Behavior Project is trying to assess how neural coding and dynamics is altered by the behavioral state of an awake behaving animal. So to do that, we're using systems developed by the Passive Viewing Brain Observatory that you may have heard about yesterday, to assess using optical physiology to assess how neural circuits are altered while an animal is behaving during a visual change detection task. So we use standardized approaches to breed mice, have implant cranial windows, identify brain regions of interest, train animals, do 2-photon calcium imaging, either of a single cortical plane or of 8 simultaneous cortical planes, and then process the tissue and develop a standardized data product. So the first thing I want to tell you about is just what is the behavioral task that we trained mice to do. So this is a visual change detection task. So a mouse is head fixed. You can see here in this video of a mouse performing the task. This is a mouse. There's an optical microscope attached to the mouse's head. The mouse is running on this treadmill that doesn't influence the task at all, but the mouse can run if it feels like. There's a lick spout that the mouse can lick, both to indicate its choice and also receive a water reward. And on the background, a screen is showing the animal images. So the task is images are serially presented in a systematic fashion that it's repeated. So in the color scheme at the very top, you can see a cartoon of what image is being presented. So the image is repeating this yellow image. Sometimes the image changes, there was just a change. The mouse has to lick when the image identity changes. So an image is being presented repeatedly. You can see here in the bottom left, the images in this image set that we're using. So for example, it might be an image of a flower turns into an image of some grass, and the mouse has to lick to get a reward when the image identity changes. Sometimes the images are omitted. I'm going to stop the video right here. So the image is presented for 250 milliseconds, normally followed by 500 milliseconds of gray screen. Sometimes an image doesn't appear. We call that an omission. Those happen randomly, and the mouse is supposed to ignore them. Sometimes the mouse licks when there is no change, and the next image change is then delayed. When an image change does happen, you can see here, there's a response window. The mouse has to lick in the 750 milliseconds following the image change. If they do respond like the mouse did here, they get a water reward indicated in this video by a blue dot, and then they have several licks where they consume that reward. So this is the task we're using. This is a visual change detection task. So we're asking where in the visual cortex does the mouse learn to detect, okay, this is a new image from what I was previously looking at. How does the sensory input interact with the persistent memory of what the mouse has looked at in the past? And how are those dynamics shaped by the behavioral state of the animal? And I'll show you our experimental manipulations of the behavioral state of the animal on the next slide. So here's our experimental design. And for each mouse, the mouse trains over many behavioral sessions where we're not recording from the brain. And then once the mouse is fully trained, we transition them to the 2-photon calcium imaging rig, where we record from 6 behavioral sessions in a row. The first 3 behavioral sessions use a familiar image set. So that's a set of 8 images that the mouse was trained on. And then we transition the mouse to 3 imaging sessions with a novel image set. These are natural images the mouse has never seen before. So our first manipulation of behavioral state is how does neural activity respond to a change from familiar imagery to novel imagery. The second manipulation of behavioral state is active and passive behavior. So on the active sessions, the mouse can lick in order to earn a water reward and do the task as I just described. On passive sessions, the lick spout is removed and the mouse isn't allowed to respond. So the stimulus just plays and the mouse has to watch. So we can manipulate how neural activity is different from when a mouse is actively doing a task versus passively viewing it. So that's our second manipulation. I already told you we have image changes, and then sometimes images are omitted. That's a violation of their expectations. So those are other behavioral modulations. When we do this calcium imaging, each mouse is expressing a transgenic GCAM6F reporter, and it's either localized in one of these cell types. So we have a pan-excitatory line, and then we have 2 inhibitory lines, SST and VIP. And the reason for recording from these 3 cell types is shown here in this schematic from a paper that was discussed in yesterday's tutorial. And this is a common motif in visual cortex where we have these pyramidal excitatory cells that have reciprocal inhibition from PV cells that we didn't record from. But then these SST and VIP cells have a mutual inhibition motif between them, and they receive input from other parts of the brain, and then they modulate these excitatory cells. So because we're interested in behavioral context, we recorded from the excitatory cells and these 2 inhibitory classes that are believed to be playing a role in modulating the excitatory cells. So at a high level, here's some of the features that are available in this data set. So first of all, there is behavioral data that is very rich and is worthy of exploration. So for example, the mice are responding to changes in image identity. 5% of all images are randomly emitted. We record the running speed, the licking data, and the pupil diameter of the mouse. We also have multi-day cell tracking. We attempt to record from the same cells over all 6 sessions. We have 3 sessions on a familiar image set, 3 sessions on a novel image set, and then active and passive behavioral sessions. We are doing cell type specific 2-photon calcium imaging, either of a pan-excitatory line, an SST inhibitory line, or a VIP inhibitory line. We're recording from 2 cortical areas in the mouse, V1 and LM. And we use 2 different microscopes to do this data set. One of them records just a single imaging plane. The other records up to 8 simultaneous imaging plane. And when we do that, it's 4 cortical depths between these 2 cortical areas. And I'll tell you a little bit more about that when we get to the tutorial. Finally, all of our data is available online. You can download Neurodata Without Border files. The Allen SDK provides a Python code base for accessing and doing basic analysis of this data. and you can access everything at this website. And I'll show you more in the rest of the tutorial about how to access the data. Before I show you how to access the data, I'm just going to give some very high-level findings of the sorts of things that we can see in this data. So first, I'm going to show you a behavioral result that the task performance generalizes to novel images. So the mice aren't phased when we change up the images on them. Also going to show you that mouse engagement changes over the course of the session. So within one session, there's changes in the animal's behavioral state. I'm going to show you that image novelty modulates the neural responses. So excitatory and VIP neurons are more active on this novel image set, even though the behavior is very similar. And finally, I'm going to show you an interesting result that VIP neurons respond to these image emissions, but only during the familiar image session. So there's something interesting there. Okay, so first, just high-level result about the behavior on this task. As a reminder, our data set contains these 6 sessions where we have 3 familiar image sessions, 3 novel image sessions. We're just showing you the performance on the active sessions because on the passive behavioral sessions, the mouse can't respond, so we don't have a performance readout. On the right here, I'm just showing you the number of hits or the number of image changes the mouse correctly responded to across those sessions. And across the population, you can see it's very consistent. There's no increase or decrease in performance when we transition the mice to a new image set. I can also, on the left here, just plot the hit rate percentage. So this is just the fraction of image changes that the mouse correctly respond to as a function of the number of images presented in the session. So this is about a 1-hour period. And you can see a pretty stereotyped pattern across all 4 active behavioral sessions where the mice perform pretty well at the start of the session, and then over the course of the session, they disengaged and they stop responding, presumably because they've gotten enough water that they're no longer motivated to do our task. So there's a lot of interesting behavioral dynamics in this data set that's worthy of study in its own right, but we also have neural data. So one obvious thing we see in the data is that novelty increases the magnitude of image evoked responses. So here I'm just showing you the population average for excitatory, SST, and VIP neurons when either showing the animal a novel or a familiar image. So excitatory neurons, this is the time of the image presentation on a gray screen. When you show the animal a novel image, you see a larger response. You also see that for VIP neurons. In SST neurons, you actually see a slight decrease. So showing a novel image changes the neural properties we record, even though the mouse behavior doesn't change. Finally, we can see that VIP cells respond to the image omissions. So as a reminder, images are being presented sequentially. Sometimes images are randomly omitted. When that happens, the SST cell or the excitatory cells don't respond because they're, for the most part, visually driven. The SST inhibitory neurons don't respond. However, the VIP neurons do respond. They have this ramping response. I'm not going to go through the data in detail, but we see this pattern only in familiar image sets. When we transition to novel image sets, these VIP cells start becoming image responsive and not omission responsive. So that's another interesting thing. So now I'm going to go to this interactive notebook, and it runs in the cloud, so you should be able to follow along if you want, but I'm going to go through it. So I'm going to stop sharing my screen and then reshare with the browser. Okay, so you should now see my browser again. So this is the GitHub repo for this tutorial. If you want to get to the notebook I'm about to show you, you can just click on Visual Behavior. And then you can just click on the Google Collaboratory Notebook. I already have it sitting here. So the first thing we're going to do is just click this button that's going to install some packages. Because everything is in the cloud, it'll take a second to run. Basically, everything's running in the cloud, so you don't have to install anything on your local machine. In this tutorial, I'm going to show you how to access data from multiple imaging planes that were recorded simultaneously during a 2-photon imaging session. We're going to construct some event-triggered plots of single cells and some behavioral data, and I'm going to show you how to track single cells across multiple imaging sessions. So that runs. The first time you run it, after you install everything, you're going to have to click Restart Runtime. That's just a particular thing about this Google Notebook, that if you're running this on your local machine, you won't have to worry about that. So we're going to have to import some packages. We're importing some general Python tools and then some Allen Institute specific things. These first two are specific to this visual behavior data set, and the rest is from the Allen SDK. So if I run this, what we're going to do is we're going to download some tables that are basically manifests of all the data that you might possibly want from this project. So for example, the OPHYS or optical physiology session table tells us what imaging sessions were collected and are available for you to look at. The behavior session table contains all the information about every behavioral session, whether or not it contained optical physiology. So that could be training sessions, or it could be imaging sessions, and you just are not worried about the optical aspect. And then we have the OPHYS or optical physiology experiment table, which tells us the data describing every imaging plane. So sometimes a session has one imaging plane, and sometimes a session has up to 8 imaging planes. I'll get into a little bit of that later. Actually right now. So there's a lot of terms we're going to be using in this tutorial. This graphic just helps describe what I mean by everything. So for one mouse, which is this purple box, we're trying to collect data for up to 6 days. Sometimes there's more if there's retakes for various reasons. Sometimes we're recording just one imaging plane, but other times we're recording up to 8 imaging planes. So we refer to all of the data from one mouse as a mouse. A container is the one imaging plane tracked over multiple sessions. A session is all the imaging planes recorded at one time. And an experiment is a single imaging plane in a single imaging session. So if you get confused about what I mean by all these terminologies, you can always come back and look at this graphic, which will help describe what we mean when we talk about different subsets of the data. So if we look at these, we're just gonna, we just downloaded the session table and the experiment table, and now we can just look at them. So if I just, these are pandas data frames, I can just look at the top 5 rows. So the index here is the optical physiology session ID, and it tells us a lot of information about what this session was. So for example, it tells us what microscope it was collected on, the genotype of the mouse, what mouse it was, what the calcium indicator reporter line was, the sex of the animal, how old the animal was. It tells us what type of session it was in that 1, 2, 3, 4, 5, 6 scheme that I told you about earlier, how many times the animal has seen that image set, how many times the animal has seen emissions. And then it also tells us the behavior session ID, if you just want to grab the behavior information associated with this imaging session. And it also tells you what imaging planes were collected as a part of this session. We can also look at the experiment table. So again, that we were just looking at the session table. Now we're looking at the experiment table, which is just each imaging plane listed separately. Contains a lot of the same information, but now it's just, there's just one imaging plane instead of up to 8 imaging planes per session. So let's just load some example data and look at it. So I have selected optical physiology session ID, that number. If we load it from the session table, we can get all of the relevant information about this session. Turns out this session has 7 experiments. So the first thing we can do is just download all the NWB files associated with those optical physiology sessions. So in this code that takes just a few seconds to run, I just clicked it. We are going to create an empty dictionary for every, we're going to load our relevant session, and we're going to get all the experiment IDs associated with it. We're going to loop through them and just using the Allen SDK, download the relevant data onto our computer. Once you've downloaded these files, they'll be local on your machine, or if you're running in the cloud, local to the cloud's instance, and you won't have to worry about it again, but it takes a few seconds to download. So again, each optical physiology session has up to 8 imaging planes. This one happens to have 7, so we've downloaded 4, and it'll take just another few seconds to download more of them. If you have any questions, you can drop them in the Q&A, and I'll be happy to answer them as we go. So once we have all of this data downloaded, the next thing we're going to do is we're going to put it together into one convenient pandas data frame. And we're going to use something called the tidy format, which if you're not familiar with, seems a little strange at first, but it actually turns out to make a lot of things a lot easier. So so we finished downloading the data. So to make this data frame where we combine all of these imaging sessions into one quickly accessible pandas data frame, I'm going to click this to run. We're just looping over our imaging planes. For each imaging plane, we're going to build a tidy cell data frame, and then we're going to combine all of these data frames together. So in the tidy format, each row is one observation of one cell. So if we record from one cell for up to 100 timestamps, then we're going to have 100 rows in our data frame. And basically, because pandas data frames are highly optimized to filter rows, it ends up being a lot faster to construct a data frame of this format. So it's almost done running here. So we can just look, how long is this data format? It's like 3.8 million rows long, because we have the number of rows here is the number of time points we recorded from times the number of cells. So it's a really long data frame, but it's going to let us do a lot of things really easy in a second here. So we can just say, what are the columns that we have now collected from this neural data? Well, we have timestamps. So this first row is an observation of this cell at this time, and its delta F over F value is this. It came from this imaging plane, from this imaging session. It was located in primary visual cortex at this depth. So one thing you might have noticed is we have 2 cell IDs. So what are these numbers? Well, a cell ROID is the unique region of interest, ROI, for all cells in a given imaging plane or imaging experiment. The cell specimen ID is a unique ID for all cells that we tracked across multiple sessions. So if an ROI is detected in 2 imaging sessions and we think it's the same cell, we give it the same cell specimen ID, but they're always going to have separate ROIs. So for the most part, we can always use cell specimen ID, and that will tell us which cells were recorded over multiple imaging sessions. So one thing we might want to know is how many cells did we just download? So we can ask how many unique cells are in our data frame. Turns out there are now 79 unique cells that we just downloaded across these 7 imaging planes that were all recorded at the same time. I'm going to pick one of these cell specimen IDs for us to look at a little more carefully. If we just query our pandas data frame to give us just the timestamps associated with that cell, we can get a data frame that's specific to just that cell. If we want to look at the neural data, we have 3 columns that are relevant. The first is the delta F over F column. So this is the change in the calcium fluorescent signal normalized to a background fluorescence. So this is a commonly used signal in systems physiology. We also have the deconvolved calcium events. So calcium indicators are this continuous signal. You can also try to approximate when the cell was electrically active by deconvolving back to detected events. You can read more about how this process is done in our white paper, which we link here. And for some analyses, detected events is more appropriate. We provide both ways of looking at the data. Finally, filtered events is just the event signal that's been smoothed by a Gaussian kernel, so it's a continuous signal. For the most part, we're gonna today just look at this delta F over F signal. So the first thing we do is we might wanna plot it. So here I'm just plotting the delta F over F signal for this one cell over a 1 hour recording session. So this is in units of seconds. You might notice a couple things. First of all, there's this background level that is kind of a noisy background, and then there are large transients. So these are large calcium events that we're picking up. You might notice they kind of start pretty abruptly at this time. That's because the imaging session has 5 minutes of gray screen, and then the task starts 5 minutes in, and so the cell starts becoming active at the start of the stimulus. I'll show you more about how to get that information in a second. So we just downloaded some neural data. This is a visual behavior task. We also want to know how to download the stimulus information that describes the task that the mouse was performing. So all the imaging planes in the session have the same session information, so we can just use any one of them. So we had this dictionary of experiments. I just picked my first experiment ID, and we just downloaded the stimulus presentations data frame. We dropped a column that's just going to get in the way for now. And now we have a data frame of the stimulus information. So each row in this stimulus table is one image presentation. Tells us how long it was presented for. It's normally 250 milliseconds due to computer jitter. There's sometimes a little bit of a difference, but it's mostly 250 milliseconds. This tells us what image it was. It tells us whether the image was emitted or not, the time it started and the time it stopped, and whether it was a change image with respect to the last image being presented. As I described, another feature of the task is the stimulus emissions. So these are images that were not presented at the regular cadence of every 750 milliseconds. So you can figure out which images were emitted by just querying the stimulus data frame for the emitted column. And this tells you all the images that were emitted. Now I'm going to show you a couple other behavioral attributes that come along with this data. So for example, we can get the licking information. When did the animal lick the lick spout? So again, it's the same for all 8 imaging planes. So we just look at one of them, we take the licks data frame, and this tells us at what times did we register a lick from that animal. We can also look at the reward information. So this is just telling us when did the animal receive a water reward. This has a column for the volume, which is the volume in liters of water the mouse received, the timestamp of when it got it, and whether it was an auto reward or not. I haven't mentioned this yet. Every session, the first 5 rewards, we give them away for free to the mouse to motivate the animal. And after that, it should be false. So these first 5, the mouse gets the water, whether or not it licked at the right time or not. So, okay, I just showed you a lot of different attributes. Let's put it all together in a plot so we can see how everything works. So in this plot, I'm going to just pick a time window of 40 seconds near the start of the session. I'm going to plot the delta F over F signal. I'm going to filter the stimulus table for images that were presented in the time window we care about. For each one of them, I'm just going to plot a colored bar. And I'm also going to do the same thing with licks and plot a little blue circle. And for rewards, I'm going to plot a red circle. And when we do all that, we get a plot that looks like this. So again, this is the delta F over F signal on the y-axis as a function of time. We're looking at a 40-second window of this 1-hour session. This colored line is the delta F over F signal for this given cell. And you can see that the cell responds to each image presentation, which is a colored line. It tends to have a little peak every time an image is presented. The image identity is shown by the color of these bars in the background. And sometimes the image identity changes. So it's one image is being presented here, then a new image, then a new image, new image, new image. Sometimes the images are emitted, as indicated here by these gaps in the colored bars. 5% of all images are randomly emitted. The mouse is supposed to ignore these. Sometimes they get tricked into thinking it's an image change. Sometimes they ignore them. The red dots are when the mouse received a reward, and the blue is the licks of them consuming the reward. Since this is the start of the session, these rewards are all auto rewards, so the mouse got them for free, but normally there's a lick associated with each reward that would happen later in the session. Okay, so that's a general overview of all the data. So just to recap what we've done so far, we picked a random imaging session and a random cell. We downloaded all the neural data associated with that imaging session across multiple imaging planes, and then we plotted a bunch of behavioral attributes that were all recorded simultaneously. The next thing we're going to do is build an event-triggered response average. So we're going to say, on average, what does this cell do in response to an event of a certain type? And we're going to look at image omissions. So basically what we're going to do is, because all of our data is in this tidy format, it's really convenient to just query it. We have this function called event triggered response that's in the Mindscope utilities package that we installed at the start of this tutorial. And if we run this, it just gives us a data frame of the neural activity with respect to the time with respect to the thing we queried on. So we queried on event times that are all these image omissions. And this gives us a trace of the average activity of this cell around the times of image omissions. So this has columns of time relative to the event, the delta F over F signal, and some other information about the image omissions. You can read, if you click this, it'll bring up the documentation for that function, and then we can just plot it. So this is going to, using the Seaborn package, creates a confidence interval using a bootstrapping approach. And here you can see time aligned to the image omission. The cell is regularly active because the images are presented sequentially. The image omission happens, the cell isn't active, and then it responds to the sequential image presentations. To make that more clear, we can just add some annotations for stimulus information. So because we know the image times, because they just happen on a regular schedule, we can add colored bars for each image presentation and just annotate the plot a little bit. So for this one cell, we're now looking at the average delta F over F activity at the time relative to every image omission. So with these colored bars for the image presentations, it's quite easy to see this cell is image driven. And then when image omissions happen, it's not active. And then it's active again for more image presentations. So we're going to explore this a little more, looking at more cells in the imaging session we recorded from. To do that, first, we're going to make just a nice function that computes the event triggered response and then plots it for us, just as a nice wrapper, so we don't have to write a lot of code over and over again. So this is going to give us the same thing we just plotted, but using our new function, except instead of using the average delta F over F signal, I've swapped out here filtered events. So this is giving us the same signal, but now using the detected events magnitude. This is just to demonstrate that you can do that. I'm going to go back to using delta F over F for the rest of the tutorial. So, okay, we previously just looked at one example cell. Let's look at more cells. So here we're just going to take 10 random picks from all of our cell IDs and plot their delta F over F average responses to image emissions and plot them together. It's going to take a few seconds to run because it's averaging over a lot of data. So when this pops up, there will be 10 traces, which is the average response of 10 randomly selected cells in a 3 or a 6 second time window around every image emission. So it's going to take about 8 more seconds. So again, there are now 10 traces here, showing the average activity of these 10 cells in response in a 6 second time window around when the image was emitted. And you can see that the cells vary both in their magnitude and what do they do in response to the image emission. And it's important to think that, not all cells do the same thing. So another thing we can do is, basically say, let's look at the average response for every imaging plane. So we're recording from 7 imaging planes in this session, split across 2 different cortical areas. Let's group by imaging plane and see if there are large differences across the depth we're recording from in these 2 cortical regions. So the first thing we're going to do is use the pandas data frame tools, group by and mean, to average across the imaging planes. And this now just tells us the average delta F over F by experiment. Again, experiment means imaging plane. And now we can use our 7 experiment IDs to average over the things, or to make the event triggered responses for the average data we just computed. So this, again, we're just gonna make a plot. We're gonna loop over our 7 imaging IDs, imaging plane IDs. For each one, we're gonna compute the event triggered response in response to the emitted images. We're gonna get some metadata about what the cortical depth was in the region and add that as a legend. And then we're gonna plot everything. So it's almost done running, a couple more seconds, and it's gonna plot up with 7 lines. Okay, so we have the same plot we've been looking at, the average delta F over F in response to the time around image emissions. And there are 7 lines here, and the 7 lines are color coded to be different cortical regions and imaging depth. So for example, this blue line here is primary visual cortex, VIS-P, at a depth of 275 microns. Over here we have VIS-L, visual LM, at a depth of 75 microns. So you can look at, say, okay, do cells in different parts of the visual system respond on average different to image emissions? You can make the same sort of plot to look at different types of cells. The session we downloaded happened to be an SST inhibitory line, but of course, you could have also looked at the VIP inhibitory line or the SLC pan-excitatory line. So there's a lot of different attributes to explore here. If you want to look at a full description of the data set, you should go to this website, which will tell you a lot of different ways to look at this data. So now let's look at a couple more behavioral streams. So we're going to just show the pupil diameter and the running speed of the animal. As a reminder, the mouse is free to run. It's on a treadmill. It doesn't have to run, but the mice tend to run quite a lot. So we can look at this running data frame. Again, it's the same for all of our imaging planes, so we can just pick one at random. And we get a data frame that's the timestamps and the speed of the animal. We can now just plot the running speed triggered around image changes. So we're using our same make event triggered plot, but we're asking to average over the speed of the animal and aligning to image changes. We can compute when, since the image changes, we're going to make the colored bars for the stimulus presentation one color before the image change and another color afterwards. So here we're showing for this animal in this session, the average running speed in centimeters per second in a 6-second window around image changes on average. So there's a couple interesting things that you can see when you look at this plot. First of all, there's this scalloping effect where the mice tend to start speed up and slow down in sync with the image presentations. And then at the time of the image changes, there's a drop in speed and then it recovers. If we were to break this down into hits and misses, you'd see that for hits, they tend to slow down and stop running while they drink their water, where for misses, they tend to keep running. So that's probably what's driving the drop here, is that the mice tend to stop running when they respond. We can also look at the pupil diameter of the animal, which is a physiological signal that may be of interest. Again, there's this eye tracking data frame that's the same for all imaging planes. So we can just download that eye tracking data frame. It tells us a lot of information about the eye area, whether the animal was blinking or not, and the size of different dimensions of the pupil. If we make an event triggered pupil diameter again to the image changes, we can see some interesting things happening. Again, there's this scalloped effect like we saw with the running that dilates and contracts with the image presentations. That kind of makes sense just from a basic reflex. If you're presenting an image, you might expect the pupil to dilate or constrict in response to that. So there's lots of behavioral streams of data. These are just two I wanted to point out. And maybe the most interesting thing in this data set is that we're tracking single cells across 6 sessions. Remember, there were 3 familiar image sets and then 3 novel image set sessions with active sessions interleaved with passive behavioral sessions. We attempt to record from the same cells over all 6 sessions. Doesn't always happen for experimental reasons. So now I'm going to show you how to track individual cells across multiple sessions to compare activity in different behavioral contexts. So the session we were just looking at had a session ID. We can use the session table to say, what was the mouse ID that we were just looking at? Okay, the session I randomly picked earlier had this mouse ID. So now we can query the session table for all sessions that had the same mouse ID. So these are all the sessions that this mouse performed. You can see there's 7 of them. Sometimes there's more or less if a session is retaken for some experimental reason, or sometimes there's less if a mouse can't continue to do experiments for some reason. The columns here tell us what experiment or what equipment it was collected on, and importantly, the session number. So I remember there are these 6 sessions, and they are described by this numerical number. Previously, we were looking at session 1, which was the first image session with the familiar image set. And now we're going to look at a novel image set. So I selected this new novel OFIS session ID that I got from this table. That is the first time the mouse was exposed to the new image set. So we're going to do the same thing we did before and download the NWB files associated with this new session. So it takes a few seconds because each one of these files is about 230 megabytes. Again, it takes a second to download, but once it's on your computer, you don't need to download it again. So it's pretty fast. This is running in the cloud, so it has to download it from the Allen Institute server to the Google instance where this notebook is running. This is a good time to jump in and ask any questions if you have any while this is downloading. I believe this, the first session had 7 imaging sessions. I believe this one has 8, so we'll see 8 files downloaded here, if I remember correctly. And once these are downloaded, we're going to do the same thing we did before. We're going to combine all of the data together into a tidy data frame, which allows us to make these event triggered plots really conveniently. So it's almost finished downloading. Then we're going to rebuild this neural data frame that is going to take another few seconds to run. Okay, so there was actually 6 imaging planes associated with that new experiment. Now I'm just going to combine everything together into one data frame. The slow step here is building the tidy cell data frame for each experiment. Because it already did it for the earlier 7, that part was quick, and then it takes a little longer to build it for the new ones. This is just making one big data frame that has the same columns as before, but now both imaging sessions are combined into one data frame that we can query really efficiently. It's almost done running. It's all done. So if we look at the first 5 rows, we can see it has all the same columns that we were just looking at. But now if we ask what are the unique session IDs, you can see there are 2 sessions here. We again need to get the stimulus information for the novel session. So we pick one of our novel imaging IDs, get the stimulus presentations table, just like we did previously. And now we want to just compare cells across sessions. So I already mentioned that the cell specimen ID refers to a cell that has been tracked across sessions. So we might just want to ask, how many cells were in the new session? And then how many cells were matched? That is, how many cells were in both the old session and the new session? We can print out all that information. And you can see there were 79 cells in the familiar imaging session across 7 imaging planes. There were 53 cells in the novel imaging session across 6 imaging planes. And there were 39 cells we tracked across those 2 planes. So let's pick a cell at random and plot it in both conditions. So we're just making an event triggered response for one imaging session and then the other. And we're going to make it in response to the image emissions. And now this plot looks really interesting. So before we made for the same plot this blue line, and we saw that the cell responded to each image presentation and then didn't respond during the image omission. But now the cell shows the same pattern, but a much larger amplitude on the novel images. So at the start in the slides, I showed you that the cells change their activity in response to stimulus novelty. We see this cell is more active during the novel image sessions. I'm going to stop it here so we have time for some questions. This tutorial goes on to compute some more things like averaging across sessions for the familiar and novel images and looking at image changes. You can follow along and run this in the cloud, but I'll stop it here and ask and offer to answer any questions. So if you have any questions, drop them in the Q&A.
Well, I think there are no questions in the moment.
Okay, well, while some questions are formulating in your head, I'll just show you a couple more features of the data set. So we had just looked at one cell in both a familiar and novel imaging session. We can compute the average across each imaging plane in the 2 sessions. So this is the plot that we showed before of the average response to each of the 7 imaging planes on the familiar image set. If we do the same thing for the novel session, then things look really different. So in the familiar imaging session, a lot of these cells were responsive to images and then stopped responding during the image omission. But now we see something curious that in some of these imaging planes, during the image omission, the cells actually become more active. So there's a qualitative change in the coding properties of these cells when we move to a novel imaging session. Again, this runs in the cloud, so you can run it on your own computer. If you want to get started with this data set, you can go to this website, which from the tutorial GitHub, you can just follow this link to come to the Allen Institute website, where we have an overview of the data set, the change detection task, and the neural data. You can read a white paper about how we process the data, or you can click on this link, download data, which gives you a lot of details about how to use the Allen SDK and how to install it and analyze the data. So it should be relatively easy to jump in and get started looking at the data.
Yeah, great. Thank you, Alex. It was very nice. So we still have some time before the next session. If people do have questions, please post them. But maybe I can ask you a couple of things, which I hope might be of interest to the audience here. So one, I think it might be useful to hear a little bit more about the technical components here of the imaging, right? So there are two types of data. One is collected with a single plane microscopy, and the other using this multi-scope approach, which allows you to do the multiple planes. I believe the data set contains a couple of modes of those which obtain the data from the different areas or potentially from the same area simultaneously. So I don't remember that piece. Maybe you can clarify that. And also, what are the trade-offs between the two approaches, because they have different sampling rates?
Yeah, that's a great question. Let me scroll up here just to a diagram. So we have two microscopes. In one of them, there's single plane physiology. We record from one cortical depth in one brain region, in either in area V1 or in LM at different depths. When you record from a single plane, you can record at a higher imaging rate. Our microscope records at 31 hertz, so 31 samples per second, which gives you higher time resolution of neural activity. If you use our multi-scope, we can record from 4 imaging depths across the two cortical regions. In this data set, it's V1 and LM. Future data sets are going to have more visual cortical regions. And you get to record from more cells spread out in the brain, but it comes at the cost of you have a lower imaging sampling rate. So this data is collected at 10 hertz, so 10 samples per second. Neural data collected with 2-photon calcium imaging varies greatly. There are people who record as low as like 1 or 2 hertz. So our multi-scope at 10 hertz is a pretty middle ground imaging rate. Does that answer the question, Anton?
Yeah, I think so. Thank you. Okay, so maybe another thing that we might discuss a little bit here is just, scientific background and framework. So yesterday we heard from Josh and Saskia, who discussed the recordings in vivo, but for the mice that do not perform any task, right? So this is a passive viewing paradigm. And there the recordings were done from multiple visual areas. So with the NeuroPixels probes from actually simultaneously from 6 cortical areas and also some subcortical areas such as LGN or LP. And in the optical physiology data set, it's a one area at a time, but there they collected data from V1 and then a few other higher visual areas. So maybe we can discuss a little bit the question of, what is the most interesting area to record from for the task like this that we are studying here. do you expect substantial differences, let's say, as we go from V1 to LM and then beyond that to even higher cortical areas? And then, I mean, this task is not just about vision, it's also about behavior, and where do you expect to see most interesting things in terms of behavior, maybe outside the visual cortex?
Yeah, great question. So scientifically, you're right that this task includes a visual sensory component, but also a working memory component. The mouse has to remember what it was just looking at in order to decide whether it needs to respond. And it also includes factors like attention and motivation, because the animal is engaged at the start of the task, and then after 1 hour, they tend to disengage and stop responding. So I think we expect naively that as you move through the brain from more sensory areas like V1 into higher visual areas and then into non-visual areas, we're going to see a transformation from sensory signals into more working memory or cognitive signals. And we've seen some hints of that. This data we've released is just from 2 brain regions, so can't say too much yet, but we have plans to record from more brain regions, kind of moving deeper into the more abstracted parts of the brain.
And what is your favorite area that you would like to record from for this task?
Well, why pick just one? Let's record from everything. That's my pick.
That's the right approach for the Allen Institute.
All right, well, should we end it there and have a 5 minutes before the next tutorial?
I think so, yeah, let's do that. So thanks. Thank you, Alex. That was very nice. Yeah, thanks everyone. So we have 4 minutes. Let's just wait for that. If people want to join for the next tutorial so that we start on time. Yeah, so we'll continue in 4 minutes. All right, well, it's 11 a.m. here in Seattle, so let us go ahead and get started. So thanks again, everyone, for joining us in this last session of these 2 days of tutorials. We'll talk about biorealistic modeling. So about the models that we've developed and are developing using all the data and some more. So the data that you've heard about earlier today and yesterday. And about modeling tools, the tools, software that allow us to do this sort of modeling, and we share all of those with the community. So yeah, let's go ahead. So I'll give some introduction here, and Kael then later will walk us through some tutorials on using the tools. And please post your questions in Q&A. We'll be happy to hear from you. So right, so over the previous day and today, you heard about this really cool data and very nice and complete data sets from the Allen Institute that characterize the components and function and connectivity within the brain. And for models, this is really a treasure trove that we are using with the idea of establishing this virtuous cycle of experiments and modeling. So we start from the properties of cells and cell types, sort of what Nathan described earlier today, morphology and electrophysiology of cells, of neurons. We use the connectivity like Luke described yesterday, the connections between cells and the synaptic properties, and the neurophysiology obtained in vivo that tells us something about the functions of the cells in the awake behaving animals. And then we put this data together, we integrate them into this large-scale biologically realistic models. We run simulations of these models, and all of that is freely available, just like this data, for everyone to use. And ultimately, we want to take these models and simulations, make some predictions about what might be happening in the mouse brain. Of course, we here at the Allen Institute primarily focus on the mouse as our model system, but of course the tools that we develop and share with the community can be used for other species too. And then, with these predictions from models and simulations, we hope to be able to design new experiments that will then result in better models. And so we go ahead that way, sort of hand in hand in this virtuous cycle of experiments and modeling. So that's the idea. And I'll tell you today about some of the progress that we've made in that area, and again, about the tools that we use to do this work. So very briefly, in terms of integrating the data that we collected, it all starts from the building blocks that are the cell types, like Nathan told you earlier this morning. We obtained information for hundreds and now thousands of cells with electrophysiology and morphology. We developed methods to build models of individual cells like that from these data sets at multiple levels of resolution, right? So we can build models of point neurons, of compartmental biophysically detailed neurons, either with active conductances restricted to the soma or with active conductances distributed across the dendrites. And of course, there are trade-offs between all of these models in terms of how much so what kind of level of detail we can reproduce with them versus the computational efficiency, as well as the amount of data that we have to constrain those models. Because, for example, we really don't have much data about the dendrites, but we have a lot of data about what happens at the soma in terms of current injections and the voltage responses. So in any case, all these methods have been made available, and the models are now available for hundreds of neurons, and one can find them here at the Allen Cell Types database. And we use them as the building blocks for our network models. So in terms of connectivity, you heard from Luke yesterday about this really cool data set from the multi-patch synaptic physiology that characterizes the connection probabilities between different cell types in the mouse primary visual cortex, but also the synaptic properties. So the weights, kinetics, dynamics, those are all very, very valuable data. We also, something that we didn't discuss over these 2 days is another new data set, the electron microscopy reconstruction, also of the mouse primary visual cortex, that's gradually coming along, and some of that is already available. So this is also a really important data set that we are leveraging for our models. So the idea here is that we take data from these data sets and integrate them in the form that allow us to build models, the network models. However, what we have currently available in terms of models couldn't take advantage of the data yet, because several years ago when we were doing this, these new data sets weren't available yet. So actually what we did so far was to integrate the data from the literature, right? So we looked at a lot of the different papers available and took the data from there, assembled that into sort of a systematic understanding to our best ability at this moment of how the connectivity looks like, and that's what we used in the model. But currently, we are making the next step and using these newer data sets to develop newer generations of these models.
Anton, quick question about connectivity. Is there any plans to do connectivity analysis in the hippocampus or other regions of the brain?
So we don't have those immediate plans as far as I know at the Allen Institute. I can't really speak for everything. The Allen Institute for Brain Science might have some plans that are taking shape gradually, and maybe hippocampus may feature there at some point. But I'm not aware of anything like that at the moment. So yeah, I would say in the near future, I think we are still focusing on the cortex, but not the hippocampus yet. All right, so yeah, so from there, there is another component that's required for the models, and that's the inputs. So we can have a nice network model, but we need to drive it with some kind of input so that the input comes in and then the model reacts to it somehow, right? And for the visual cortex, which is what we are focusing on, the primary pathway for providing the visual information goes from the retina to the thalamus, the lateral geniculate nucleus, or LGN of the thalamus. And from there, spikes from the LGN go into V1, which is what we are modeling. That whole pathway is very complex, and we don't have enough data to really model it in a very high level of detail. So instead, we replaced it by this filter layer, which takes as an input movies, right? So three-dimensional X, Y, and time arrays of data that tell us what appears sort of in the visual space. Then each filter here represents an individual LGN cell, which processes that input and turns it into a spike train. And the spike trains then impinge on the neurons in our V1 model. So this is the model to drive the network. So we had data for recordings from the LGN in vivo, and we optimized this filter models based on those recordings, coming up with 14 different types of LGN responses that are modeled by these filters. We have about 17,000 of these LGN cells, just like in the mouse LGN, providing inputs to our large model of V1. And we are aware that these filters are not perfect. They do not necessarily reproduce all of the aspects of LGN activity, although for some things they are doing pretty well. But the nice thing about using the filters here is that they can be used to process arbitrary visual stimuli. So any movie, and that means that we can take any visual stimulus like drifting gratings or static gratings or natural scenes, natural movies, locally sparse noise, anything else that our colleagues, experimentalists, are using, and run those movies for our model, right? Simulate our model with that specific input so that we then can compare apples to apples between the model and the experiment. And this works pretty well. Of course, there are limitations, again, just how well those LGN filters approximate the actual LGN activity, which is important and something we would like to improve eventually. And also you can consider a limitation in terms of compute power, of course, because it depends on how long your movie is, whether it's practical to run that movie through this all these mathematical objects or not. But in principle, you can use any arbitrary movie to stimulate your simulation. Okay, so with all of that coming together, we now have these models that we released a couple of years ago, and then published a paper about it last year. So the models represent this majority of neurons in V1, focusing on the central portion of V1 of the mouse brain. They represent all the layers from layer 1 down to layer 6 of this cortical area, divided into 17 major cell classes. So a lot of sort of biological realism and diversity in terms of cell classes here, over 230,000 cells altogether. And we have two versions of this model, a biophysical version and a point neuron version, where in the biophysical version, the core of the model, the central 50,000 neurons, are represented using this compartmental biophysically detailed representation. And then the rest are here in periphery, basically represented as leak integrating fire point neurons, providing or taking care of the potential boundary artifacts. And then in the point neuron version, it's a one-to-one correspondence between the two models in terms of the cell locations and the connectivity graph. But of course, the point neuron version is much faster to run. So the biophysical version is much more detailed. It can be used perhaps for a more diverse set of applications. On the other hand, we found that in terms of spiking dynamics, the two models perform quite similarly to each other. But the biophysical version is much more expensive computationally. You need hundreds of CPUs to run it, maybe get about 1 second within 1 hour. Whereas for point neuron version, you get 1 second simulated in minutes, and you can run it on a single CPU. And the good thing is that the models are freely available. Again, here on this website, you can come and download the models, look at the methods, and the results of the simulations are also there. Okay, so now we don't have much time, and we want to get to the, going through the hands-on exercises and the notebooks. But I'll just spend a couple of slides to talk about what we do with these models very briefly, and then details, of course, can be looked up in our paper. But the basic thing is that one can compare with the experiment the responses of the model to, let's say, visual stimuli, right? So in the experiment, we can show a mice, two mice, we can show some visual stimuli such as drifting gratings, for example. We can record, let's say, with the neuropixels probes, the spike in dynamics in response to this stimuli. And then we can do the same in the model. So here's an example with a model running first half a second with a gray screen, and then the rest of the time with a drifting grating stimulus. We see responses here sort of for the core portion of the model with the 50,000 neurons there across all the different layers for the different types of neurons in different layers. And now we can compute some statistical properties of these responses, of the spike in responses, and compare them between the model and the experiment. So we've done quite a bit of that, and here I'm just showing some examples. And the important point is that we don't want these models to be constructed and used for just one application, to answer just one question. We view them as multi-purpose models, right? So ideally, you will be able to do a number of different computational experiments with them, use them to deal with a variety of different hypotheses, variety of different observables. And for that, we need to be able to reproduce different features of different stimuli, for example. And this is what we are doing here, just with some examples. So we can look at drifting gratings, and we can look for each cell, for example, at the rate of the response of that cell to a preferred grating, or to the direction selectivity of that cell. We can compute the statistics or distributions of those properties of individual cells across all the cells in a given population. And then we can compare between the biophysical model and the point neuron model and the experiment, right? And this is what's plotted here for the different layers and for the different populations within the layers. And yeah, we can see how well the comparison turns out to be. And then importantly, we can do it for other things like, for example, here's a full field flash. And for that, we can compute different features, such as, for example, signal correlations or time to peak to respond to this full field flash stimulus. Or we can look at natural movies as some additional features there. So the point is that this can be done, and we did it for a variety of different stimuli and variety of different responses. We can do that systematically. In many cases, we see pretty good agreement between the models and the experiments. In some cases, we see some disagreement, like here, or even more stronger disagreement here. And well, these are also good things to know, because that points us to where we should improve our models in the future. And that's certainly something that we are pursuing in the next iterations. All right, so now, again, just very quickly to give you an idea of what other things can be done with these models. One very useful thing to do is perturbation, right? Perturbation can be a very powerful technique in modeling, and of course in the experiment too. It can be compared with a variety of different experimental techniques, but the first perhaps that comes to mind is optogenetics. And we can do that relatively easily with our tools. And Kael will illustrate that later. But here's an example of the point neuron model simulated with response to a drifting grating that starts at 500 milliseconds. You see that neurons here are happily firing in response to this grating. And then we can do a perturbation. We can make the perturbation happen sort of at some point in the simulation that's described in the config file of the simulation, or we can do it in a separate simulation right away from the very beginning, which is what I've done here. So here, layer 4 excitatory neurons were completely silenced. And as you can see, that has a dramatic effect on the response of the model. All the layer 4 neurons are completely silent now, but also all the other neurons are severely suppressed, which is not surprising because the main input from the LGN, main visual input into the system, comes into the layer 4. So if you remove layer 4 responses, then there is much less drive to all the other cells here. Okay, so we can do those sort of things, perturbations. We can also compute responses of the models that are not necessarily spiking responses. And that's especially interesting in the case of the biophysically detailed model. So here's an illustration of our model with this, a lot of neurons with their dendrites. And then we can imagine that we place a probe, like a neuropixels probe, with multiple electrodes that go through this cortical depth. And we can represent those electrodes in the simulation and ask what kind of electric field they record. Of course, in principle, we can keep track of every single compartment in the simulation and every single current in that compartment and ask what kind of electric field they create outside of the cells. And then we can sum them up to obtain the extracellular electric field, filter it down to the local field potential, and use existing estimates to obtain the current source density, which we then can compare to the experiment. So this is just an example of how this can be done. This is work together with Atle Rimehaug and Gaute Einevold in Norway. So they are using our model and this ability to compute the local field potentials and current source density. And then, we can plot things like that, where we look at the current source density as a function of time across the depth in our model. And we can compare the different features, the sinks and sources of this pattern with the experiment and ask how the different structures, circuit properties, and cell types in the model give rise to these different features. So these are some examples of what can be done with the model. And now I'll switch gears and talk a little bit about the tools, and then Kael will give you the demonstration of the tools. So to make all of this happen with modeling, we developed some tools that for us make our life easier, and then we decided to, of course, share them with the community as well, hoping that people might find them useful as well. And so the first tool that we have is the Brain Modeling Toolkit, or BMTK. Like everything, it's freely available. In this case, you can find it on GitHub. And BMTK is a Python-based software package for building and running simulations. So this is a schematic of the modules of BMTK. It has a builder module that allows you to build models at different levels of resolution. And then it has the simulator modules that allow you to run simulations at different levels of resolution. We are not trying to reinvent the wheel here. We are not trying to replace the wonderful and very powerful tools like Neuron and Nest. What we are trying to do is to provide an interface, a convenient user interface, so that you don't maybe need to learn all the details about the environment like Neuron and Nest, but still can run pretty sophisticated simulations. So for example, you don't have to be an expert in parallel programming under Neuron or to know how Nest does it, but BMTK already provides this automatic support of parallel simulation and all the necessary code to run such simulation under these engines. And of course, we are not the only ones who are doing this. There are tools like NetPine, for example, that also do that quite efficiently with Neuron. Perhaps one I think about BMTK that I want to mention is that it does work at this multiple levels of resolution, and it provides a similar user experience under these different levels of resolution and working with the different simulation engines. And one way we achieve that is by using the file-based workflow with our models. So instead of using code all the time to represent models, to build them and then to simulate them, we separate these steps and we use files to move around components of the models. using this scalable open network architecture template, or Sonata data format. So this is a data format that we developed together with the Bluebrain colleagues, and then also together with additional partners from the open source Brain and NeuroML and Pine, NetPine, and Neurodata Without Borders. So we all came together to finalize this format and to write a paper. And I believe that it's a pretty useful format that I think is relatively widely used in the field now. So the key point here is that this format reflects all the components of the modeling workflow, right? So in a modeling project, you might have your representation of the network with nodes and edges between them, with some external nodes that supply inputs and the internal nodes that you are simulating. You are running a simulation, you produce an output, and Sonata reflects all of the steps so that you have representations of nodes and edges in CSV or HDF5, actually in both usually, especially for very large files. The key benefit of Sonata is that it provides you the opportunity or possibility to save these very large portions of your model in efficient binary HDF5 format. Then it actually, again, it doesn't reinvent the wheel. It interacts with the existing formats such as NeuroML, JSON, SWC for representing sort of lighter components of the models that don't take so much space, and where the text-based format is quite appropriate. For the simulation, there are config files that describe all the properties of the simulations, all the important constants. And then it also has a component for the output for spikes and time series, either for neuronal components, compartments, or even for not neuron dependent time series such as extracellular voltages. So all of that again can be saved typically efficiently in HDF5 file format. So the specification and the tools to work with Sonata can be again freely accessed here on GitHub, and we welcome people to use it and give us feedback. Nice thing is that because there's a whole partnership came together here to work on this, Sonata is already supported by a number of tools, of course by BMTK, our tool, but also by additional tools like NetPine and Pine and NeuroML Lite for both network structure and then for the simulation output as well. We also have a converter for the neurophysiological data, such as spikes and time series, for the conversion from Sonata to NWB and the other way around. So finally, I want to mention something new, which is the visualization tool. So this is a tool that we call Visual Neural Dynamics, or VND. And this is a tool developed by our collaborators at University of Illinois, based on their molecular visualization software that's very, very powerful and has been developed for 25 years and offers very impressive graphical capabilities. So we felt that there are some software for visualization in neuroscience, but usually it's really hard to visualize very, very large systems efficiently. And so here we rely on this visualization engine from the molecular visualization software. But now together, so with our partners, they developed the tool to use those engine for neuroscience applications, right? And this is this new tool called VND. Just recently, we had the first release of it, and it's now freely available here on this website. So this tool already got a second place in this recent competition of the Brain Initiative called Show Us Your Brains. This is an image generated by that tool, which got the second place. And basically what this tool can do is it can read Sonata files, it can provide us efficient 3D visualizations on regular computers, but also on specialized hardware. So it's very flexible in that sense, which is important. You can do it sort of as a regular user on your own workstation or even laptop, or you can use it on a very powerful graphical specialized hardware. and it can create movies of neural activities like this. So it's in early days. We just released this tool, and there is a lot more that we want to add there, but it already can do some useful work for you. And again, please use it and give us feedback. All right, so to summarize, we have multiple resources here in terms of modeling. So models are integrating the data that we were talking about this couple of days, and they're freely available. And the software is also freely available, the suite for modeling, the file format for modeling, and now also visualization tool. And in terms of models, we have models of individual cells in the cell types database and the network models here on this webpage. So we hope you use them and you find them convenient. All right. So with that, I'll thank our founder, Paul Allen, for his vision, encouragement, and support, without which none of this would have happened, and all the teams in our institute, which do all this amazing work. And now let us turn to Kael, who will speak about the actual exercises and will walk us through some notebooks. So thank you. And Kael, take it away.
Okay, I assume you can see my notebook? I
can, yes.
Okay, great. So my name is Kael. I am the primary software developer for the Brain Modeling Toolkit, or BMTK, and I'm just going to go over sort of a high-level overview of some of the core features of both BMTK and the Sonata data formats. If I go too fast, or if I'll probably skip over some parts of this, but if you want to follow along at home, you can download this or use this download tutorial from this link, which is at our BMTK GitHub repo under the documents /tutorials, where you can also find other tutorials, documentations, examples. So first step we want to do is to actually install the BMTK Python libraries. You can download and install the latest version from GitHub, or we also have a BMTK package available through Piet and Anaconda. Parts of this tutorial will also need to use Yale's Neuron simulator libraries. You can download Neuron from their homepage, or you can install Neuron using, once again, Anaconda or Pip. But what we do have is a Docker container to run BMTK, as well that will contain all the prerequisites needed to run BMTK, as well as it even includes Jupyter Notebook. So if you want to follow along at home and you have Docker installed on your machine, what you can do is you can do a Docker run of the Allen Institute /BMTK container, and you can even spin up your own Jupyter Notebook instance and just follow along this tutorial at home. So the goal of the tutorial I'm going to do now is to build a small toy network of the model of the layer 4 of the mouse primary visual cortex and show how we can simulate it across a variety of different conditions. So the actual layer 4 model, which can be downloaded from the Allen Institute data portal, contains 45,000 cells. And like Anton said, to run it, you'll pretty much need access to a multi-node cluster to build and simulate and run. This 300 cell example here, which we're going to build, will typically be small enough to run on any modern computer or laptop in a reasonable amount of time. But before we can actually run the simulation, we must first create the Sonata network files. And we can do that using the network builder component of BMTK. So either in a Python script or here in Jupyter, we can start by importing bmtk.builder.networks.network builder, and we'll go ahead and instantiate a new instance of a network, which we'll call V1 here, just for primary visual cortical area 1. We can give it any name we want, really. So essentially, the first step of building our model is going to be determining what cells, or cell types we want to have, where we want to place our cells, what properties we want to use. So in the Sonata parlance, we don't actually call them cells. We call them nodes, and not cell types, but node types. But once we sort of had this basic fundamental foundation of our network, we can go ahead and use the network builder add nodes method to actually add our nodes or cells. So here what we're going to do is we're going to call v1.addNodes to add 80 SC and 1A type excitatory neurons to our model. So when we add it, we will pass in a bunch of parameters. So we have these Sonata reserved keyword parameters like model type, model template, X, Y, and Z. And since these are Sonata reserved keywords, it means that other Sonata compliant tools will be able to analyze, instantiate these networks. However, we also have some other parameters that are not Sonata reserved keywords, but they're going to be like our custom user parameters that are going to be pertinent for explaining cells in our models. So BMTK allows us to create our own parameters, our own attributes to best explain our model. So for instance, we are going to have this tuning angle property here. Tuning angle, which is a value between 0 and 360 degrees, is a very useful attribute in describing the behavior of excitatory neurons within the mouse visual cortex. But for other cell models or other models of areas of the brain, you wouldn't necessarily have tuning angle. You might have your own special other attributes. So like I said, both Sonata and BMTK sort of allows us to make the model as complex and descriptive as we need it. So here we call it, after calling vnodes, that these 80 cells for SCNA one types, we'll call it 2 more times. So we'll call it v1.addNodes to add two more times to add 2 more populations of excitatory type of neurons. And then we'll call it one last time to add a population of inhibitory parvalbumin type neurons. So with these 4 calls to add nodes, we've added our 300 individual cells across 4 different what we call cell types or node types, and it will have it so that 80% of these cells are excitatory, while 20% are inhibitory, which is what you would typically expect in a model of a mouse cortex. So now that we've actually added our cells, or our nodes as we call them in Sonata, we can go ahead and start creating our synapses, or what we call in Sonata our edges. And we can do that using the addEdges method. So when we call v1.addEdges, the first thing we're going to do is pass in either a list or a dictionary of conditions to determine what subset of cells we want to use for source cells, and what subset of cells we want to use for our postsynaptic target cells. So here, when we call v1.addEdges, we're going to, for our sources, we're going to use this EI property. That is a custom user property to define cells as either excitatory or inhibitory, to create connections that connect all the excitatory neurons to all the inhibitory neurons like this. Once again, we'll include some reserved Sonata keywords like syn weight or synaptic weight and delay that we'll use during the instantiation, but we also, as with nodes, we can create our own custom attributes and properties. Now, when the network gets built, it's actually going to create a directed multigraph. So which means between every possible source and target cell, there can be 0, 1, or more actual edges between each source target pair. So what we're going to do, we want to use is this connection rule parameter to determine the actual number of connections between each possible source and target. So for these excitatory to inhibitory type of edges, we are passing in a connection rule, a scalar value of 1. And what this is directing it to do is just for every possible source target pair, each excitatory cell synapses exactly once with each inhibitory cell. Similarly, we'll also call addEdges one more time to create connections from the inhibitory cells to the excitatory cells. We'll also, some of the parameters are going to be different because they're actually different types of connections. But here we're also going to use a connection rule scalar value of 1. For the connection rule, besides just passing a scalar, what we could also do is we can pass in either a matrix or an array of the appropriate length. Or even better yet, what we can do is you can create custom functions to intelligently determine the number of edges between each source and each target. So here we're call add edges to create the edge connections between all the inhibitory to the inhibitory cells. And what we're going to do is we're going to create this custom connection function rule that called ignore auto synapses that will, for each source and target pair, the first thing this connection rule is going to do is it's going to look at the node IDs of each cell, and if a cell is if the source and the target are actually the same cell, it will have 0 edges. In BMTK and Sonata, cells can connect to themselves, but in our cortical model, we just want to prevent that. And if the cells are different, we'll just randomly choose between 1 to 5 connections between each possible source and target pair. So all we did was we created this easy function that for each source and each possible target, we'll just return an integer value indicating the number of actual edges between them. And then to use this connection rule, this connection custom function, all we have to do is pass in the name of this function into the connection rule parameter. And when the network model and the connectivity is getting built, it will BMTK will recognize this and use it for building the connectivity matrix. Similarly, we'll do something similar for excitatory type of edges, but for this one, we'll make it a little more complex. Here we have this connection rule, which is going to determine the number of connections based on this tuning angle property. So like I said before, tuning angle is a property of all the excitatory cells in our model. That's a value between 0 and 360 degrees. So what this function is going to do is it's for each source and possible source and target pair, it's going to take this tuning angle property of the source and the target. It's going to calculate the distance in degrees, and using the distance as a weight, it's going to sort of create a rule that cells which are closer in tuning angles, and otherwise they're more likely to respond to drifting angles of similar degrees. It will make it so that cells that have closer tuning angles are more likely to connect to each other, which is sort of a rule that can often be seen in the mouse visual cortex. And so once again, we just create this tuning angle function. It takes for each source and target, returns number of edges. We pass in the tuning angle to the connection rule parameter. And then later on, when the actual matrix gets instantiated and built, it will use this to create the connectivity matrix. So just briefly go back what I did. We instantiated the V1 model we instantiated a new network called V1. We called add nodes 4 times to add 4 different population of cell types. And then we called add edges to add 4 different edge types or synaptic types, if we want to call them. And after these 8 function calls, we can go ahead and build and save our network just by calling the build and save methods of the network builder. Here we're going to direct it to actually save the network in a folder called network. So if you run this at home, it might take 30 seconds to a minute, sometimes depending on how big it is in your machine. But when it's finished, it will create this network folder. And what it's going to do is after it's saved it, it will save these Sonata network model files that we're going to call it V1 nodes. And also we're going to have these V1 to V1 edges Sonata model files. And so these are the actual model that we're going to use to instantiate and simulate. Also, if we wanted to share them or use another tool to use them, this is the files we would use. But before we can actually simulate these network models, we're going to also build another network, which we're going to call the LGN. So the LGN is a part of the thalamus that provides much of the actual input from the retina onto the primary visual cortical cells. So we'll, as before, we'll just instantiate a new network, call it LGN. We'll just call add nodes twice to only add 100 cells. And when we add the edges, we'll just connect the LGNs directly on. The LGN cells won't connect to themselves, but they'll just sort of synapse onto the V1. And that way, during the simulation, we'll use these LGN network to actually drive our full simulation. So we can drive it using sort of synaptic inputs of our V1 model. Then when we build and save the LGN, we'll also see it's going to it'll save the new network files called LGN nodes and LGN to V1 edges files. One thing to notice is that here, by default, BMTK will sort of save all the network files individually. And one of the things that Sonata and BMTK allows us to do is actually build parts of a network piecemeal or independently of each other into different files. And then we can later on, I'll show you how to sort of connect them up. So that way, if we wanted to build more complex models or even add higher visual areas, we can build them independently without having to rebuild the V1 model itself. One other thing to notice about the LGN cells, just briefly mention, is these have model type virtual. While if you go back and look at the V1, the model type was biphysical. So virtual is a special designation in Sonata to indicate that these cells are not biophysical or even point cells. They're essentially just placeholders for either presets voltages or placeholder for spike trains. Virtual cells can't be recorded to, or they can't be synapsed onto, but they can be used to synapse and drive or biophysically detailed cells. So now we have our actual, we actually built our network files. We want to go ahead and start the simulation. As Anton mentioned, BMTK includes a number of sub-simulators, if we want to call them, for simulating across different levels of resolution. For our V1 cells, we want to use a simulator that can work with multi-compartment biophysically detailed cells. So that's where that's the BioNet simulator of BMTK comes in. If we were starting from scratch to actually run the simulation, we would need to do some setup, like create some import and download some external files, as well as create configuration files. There are scripts that help us do this sort of automatically and can help us take care of most of that. But if you're just using the notebook at home, all the components, external components and configuration files are already set up for us in the notebook. Pretty much everything what's going to be needed to instantiate and run a simulation is going to be saved in a Sonata configuration file. Here we have this file called config.bionet.json. It's a simple JSON file, so we can open it up in our favorite text editor we want. And then we can sort of see how it's divided into certain subsections that's going to direct how to actually instantiate and set up our simulation. So I'll just quickly go over this. First major section we have is going to be called networks. This is where we specify the location of our LGN and V1 network files that we just previously created. As I mentioned before, BMTK and Sonata allows us to build different parts of a network independently. So if, like I said, if we wanted to run a simulation without the LGN, we can remove that. Or if we wanted to add higher cortical areas, we can do that very easily just by making modifications to this configuration file in the text editor. Next, we have the components section. This is where we have the location of third-party files like SWC, morphology files, or NeuroML dynamics template files. Here we have running conditions, and this is obviously the run time conditions for our actual simulation. The most pertinent attributes here are going to be t-start or time start, time stop, and the time step dt. So here we're directing the simulator to run it for 2000 milliseconds or 2-second simulation. Next, we have the output sections as well as the report sections. So this is where we specify what we want to actually record, what variable, when we run a simulation, what variables we want to record, and where we're going to record them to. So by default, BMTK will always try to capture the spike trains of all the cells. Here in the output section, we can specify we want to save our output into a folder called output, and the spike trains of all our cells are going to be saved into a file called spikes.h5. We also can do more advanced reporting besides spike trains. So here we also want to specify that for a certain subset of our cells, or just for our SCNN1A cells, we want to record the membrane potential or the V variable from the soma of all these cells. So to do that, we just have to add a new subsection to reports like here. And then finally, lastly, we're going to have our input section of the config file. So this is where we specify how we want to actually stimulate our simulations. So we can, if we wanted to add any current clamps, a voltage clamping, or even extracellular stimulus, we can add them in the inputs here. As I mentioned before, the LGN cells are essentially placeholder cells for preset spike trains. In this case, the spike trains file is going to be a Sonata spike train file called lgn spikes.poisson.h5 that will be used to show when each individual LGN cell will spike and when it's and drive the V1 cells. Here's an example of using BMTK to actually generate one of these Sonata spike trains files. Here, what this small portion of code does is for each 100 LGN cells, we're going to have it fire using a Poisson distribution, firing at a rate of 8 Hertz for a 2-second duration. So now, with all that together, we've built our net model files. We set up the config as we needed to. We are actually ready to go ahead and run the simulation. You can run it in a command line using the run bionet.py script. You can use MPI, like Anton said. If you use MPI to run it on a multi-node cluster, it will take care of parallelization for us. Or for a modest network like this, we can run it in a Jupyter notebook here. I won't run it because it takes usually between 5 to 15 minutes to actually run the full simulation, depending on your machine. But what it will do is when you run this, it will instantiate the network, run the simulation, and save the outputs. So the outputs, it will automatically save for us as we specified in the config. We're saving both spike trains under a spikes H5 file, as well as the membrane reports. And here we can see the spike train raster of all our 300 V1 cells when we drive it using this simple 8 Hertz Poisson synaptic inputs. So we can plot both in the individual cells as well as so we can see the population of cells. And then here is our visualization of the membrane potential traces. So these are for all 80 SC and 1A cells. The gray are the actual traces, while the blue are the averages. Now, if we wanted to run the simulation again under, say, we want to use different inputs or record different stuff, all you need to do is make modifications to the config, rerun this scripts or piece of code before, and then it'll automatically save the results as before. So as Anton said, one thing we're interested in the Institute is doing perturbation studies. We can sort of simulate that by using adding a current clamp onto a subsection of our cells. So in this case, to do a perturbation study by using current clamps, what we're going to do is we're going to look at the input section of our config. We're going to add this new section called inhibitory perturbations. What this is going to do is it's going to create a large depressing current clamp on all our NR5A1 cells, starting at 1500 milliseconds. It will start at 500 seconds, and it will apply a large depressing current clamp. And then once we've taken this subsection, pasted it into our BioNet config, we just have to rerun it as before. As before, everything will be instantiated. It will run and it will simulate and save the results as it will save our new results. So here, just by simply making a few modifications to the config, we can see not only were we able to completely silence the NR5A1 cells after 500 milliseconds, we can also see the effect it has on the other cell populations, which is always interesting to us. Another thing we do a lot with our models is looking at extracellular field potentials or local field potentials or current source densities. So a feature of BioNet is the ability to sort of simulate what it'd be like to stick in neuropixels or really any sort of multi-channel extracellular probe into our simulation. So to do that, we'd have to make some changes, add some stuff to the report section. So here we have this ECP or extracellular potential report to record that will simulate what it's like to stick this extracellular electrode into our simulation, and it will save the membrane potential voltage, extracellular membrane potential voltage in a file called ecp.h5. So as before, all we need to do is sort of copy this, paste it into our config. We run BioNet exactly as before. And what it's going to do is when it finishes, it's going to create this file called ecp.h5. And just to visualize it, so here's what our extracellular traces for this 10-channel electrode is going to look like for our two-second simulation. So in general, there's a lot of other features and stuff that can be done very easily just by modifying a configuration file in a text editor. There are, however, going to be some more advanced features that can't be easily done using just in a JSON file. There's going to be stuff like if we wanted to, a common thing we're going to do is like when we want to adjust synaptic weights, either right before or during the middle of the simulation, like if we wanted to add something like spike timing synaptic plasticity, it would require a bunch of extra programming. But BMTK does give the opportunity to allow people to modify the code and modify the simulation without actually having to go into the dive in deep or make changes to the BMTK source code. So I don't have enough time, but here's an example of how you can dynamically adjust synaptic weights very simply. For more information about doing the more advanced stuff, I recommend people check out our GitHub and documentation page. So to get these simulations we've been running before, we were just using them to drive. We were driving our V1 model using a simple Poisson distribution stimulus. While this can be useful for discovering fundamental properties of our network, sometimes we want to create and run it ourselves using more realistic input, particularly if we wanted to compare our network with actual cell data, like data from the Allen Brain Observatory. And this is where the FilterNet simulator comes in to play. So what FilterNet is going to do is it's going to play a movie or really any visual stimulus onto a field of neurons, in this case, onto our 100 LGN cells. Based on the spatial and temporal dynamics of the stimuli, it's going to generate spike trains for these neurons. And then we can use these spike trains to rerun our V1 simulation and see how it compares to actual real data. So running FilterNet is very similar to running BioNet. Pretty much everything is going to be set up in a Synaptic JSON config. Here it's called config.filternet.json. We look at it, we can see it's similar to the BioNet config, only much simpler. For the network, we're only going to run it using the LGN nodes. The inputs is where we specify stuff like our visual stimulus. We could place a movie, a custom movie, or our FilterNet also has some pre-built stimuli like spontaneous noise, drifting gratings. Or here, we're going to do a full field flash. So we're going to have a stimulus that's going to be starting at 500 milliseconds and then at 1500 seconds. It's going to essentially blacken out the screen for a moment of time. And then lastly, we'll make a few changes to our outputs just so we aren't overwriting our previous results. Here, we're going to save the file. We're going to save the output spike trains under spikes.flash.h5. So using this FilterNet config, we run, we rerun our FilterNet simulator, either in command line or in a notebook here. And like our BioNet, it will automatically save our spike trains. So here we can see our spike trains of our full field flash for these. And I did mention this before, but our LGN cells consisted of simple on cells and simple LGN off cells. So we can easily see how turning on and off the screen affected these two populations of cells. And then just to bring it all over again really quickly, we can go back to our BioNet config. We can replace our Pazon.h5 with our Flash.h5, rerun the V1 model, and then we can see how our V1 model reacts to more realistic stimulus and use it as comparing. We can use it to compare like to the Allen Brain Observatory. So there are more features. I'm running out of time, but I would just recommend people check out our GitHub repo. There you'll find more documentations, tutorials, and examples. And if you have questions or even if you have suggestions, we're always looking to improve and add new features that we haven't thought of. Please feel free to contact me and Anton. So with that, I will give it back to Anton to finish up.
Yeah, great. Thank you very much, Kael. And thank you, everyone. I mean, we are pretty much at the time, but happy to stick around a couple more minutes and answer some questions. So there is one question right now from Mehdi. In the case of not having access to the supercomputers, how we can run the V1 biophysical model? Is there any online platform that supports the BMTK?
Neuroscience Gateway, that's a good opportunity. Older versions of BMTK are installed there. Other people are using it to do some, I don't think we've, do some large network simulation. We are looking into some other opportunities through Google, through allowing people to run these large scale simulations. But please feel free to, ask us more. Neuroscience Gateway is the best answer at the moment.
Right. Yeah. And in case you haven't heard of it, Neuroscience Gateway is this initiative which provides free supercomputer time for researchers in neuroscience. I mean, it's not unlimited. There are some it's like you can get some kind of allocation. But ultimately, yeah, that's a possibility. The thing that I don't know is how it works for people who are not in the US with Neuroscience Gateway. I think it might or it might not work. I'm not totally sure. So that's something I will need to check. All right. Another question from Thomas. Can you make this tutorial available for download? I don't see it in the download folder.
Yeah, I'll post the link, but it's on our GitHub repo.
So yeah, I'm not sure. Maybe Thomas, you were referring to the webpage that we had for these CNS tutorials. There might have been some kind of yeah, miscommunication maybe, and this tutorial didn't make it there. So we will make sure that it will, but also just go into the BMTK GitHub. You should be able to find it there. Right, Kael? It's there. Okay, another question from Mehdi. How this model can be sensitive to different orientation of stimulus, drifting gratings with different orientations? How can we select the LGN filters in a way that specific cells with similar orientation tuning respond to the stimulus properly? So Mehdi, this is this is a scientific question, right? How you put together the inputs from different LGN cells to provide to provide the stimulus to the V1 cell so that it responds with particular properties of orientation and direction selectivity, and also how the recurrent connectivity between the cells in V1 support their ability to do so. And we had a particular implementation of that in our V1 model. It's all documented in our neuron paper, so I encourage you to go to the neuron paper and check the details, which which are in the method section and also in the throughout the text. But basically, there are several mechanisms there that people have found, and so we followed that literature to implement those mechanisms in the model. It basically has to do with the convergence of the inputs of similar type, like for a cell that prefers direct movement in the horizontal direction, there will be convergence of inputs from other cells that prefer similar stimuli onto that cell. And for the LGN, it has to do with the transient and sustained responses, and then offset between the transient and sustained responses of the LGN cells. But in any case, all these details are described in the paper, so please check it out. Let us know if you have any questions after that. But also keep in mind that this is just one possibility, right? These are the questions that are not fully resolved yet in the field, and our model implements something that's working, but there might be alternatives here. All right. Yuhan Lee, could all these tools be connected with popular deep learning libraries so we can study the various high-dimensional dynamics in simulated networks? Well, I think they can be. It would require work. Right now, we ourselves here at the Allen Institute, we didn't have a plan to do that. I actually know that other people have been looking into that. So I know in the lab of Wolfgang Maas, they actually have been porting some of our models into deep learning libraries. So there might be something at some point that one could use. So yeah, it's possible. No specific plan at the moment, but probably it will happen. And another question, can we assign tuning angle to inhibitory cells too? Do you think it's physiological? Yes, absolutely, we can. And in fact, we do. So I think what Kael showed today is a sort of a toy version of the model, where we take a relatively simple approach, but in our complete V1 model, we do assign tuning angle to inhibitory cells. And different types of inhibitory cells have different types of responses. Some are more tuned and others are less tuned. So it certainly makes sense to use that for those that are more tuned. And for those that are less tuned, you also can use it, but just make the tuning very broad. So that's totally okay. And yeah, absolutely possible and physiological. All right. Kael, do you have any last comments before we go?
I would just say, if you're interested, we're always looking for feedback. We're always looking, if something we don't do yet, please contact us. We're always trying to improve it.
Yes, tell us which features you want, and we'll see if we can implement them. All right. Well, thanks, everyone. Thank you very, very much for sticking around with us. It was great. I hope it was useful also. So have a great day or night, and stay in touch. Let us know your experience with our tools and models and data. Okay. Thank you. I'll see you next time.